Precision medicine aims to match patients to therapies tailored to their unique biological characteristics. To achieve this, doctors rely on diagnostic tests that provide vital insights into each patient's individual traits. The complexity of these tests, combined with varying regulatory requirements, can create challenges in determining if different tests measuring the same marker yield consistent results. This variability makes it difficult for healthcare providers to personalize treatments and provide the best possible care to their patients. Friends of Cancer Research is stepping in to establish unique partnerships that assess and mitigate diagnostics test variability to inform policy and improve consistency in health outcomes for everyone. Our collaborations provide important insights into the performance, accuracy, and consistency across diagnostic tests to shape future policies and regulations. At Friends of Cancer Research, we acknowledge the complexities, harness the potential, and champion the power of diagnostic tests to support precision medicine. As health technology continues to rapidly evolve, our work becomes even more critical. Together, we're committed to untangling the complexity of diagnostic tests to improve patient care.
Good morning. Thank you for joining us in person for this public meeting. We would have had more people here, but as you know, there's a pause in the government, so hopefully they're on their Zooms watching us because we have been working with both FDA and NIH on this. Today's meeting will be an annual event focused on driving innovation and the diagnostic space. We don't expect this meeting will solve all the issues, but we expect that we'll be working on this for a while and working on this together. Through collaboration, we are addressing complex challenges and pushing towards the policy for all patients. I don't think there's a patient who gets a test or gets their diagnostic results who think they're not accurate or to ask if they are accurate. I can tell you myself, recently I went through an experience with cancer, kidney cancer, and getting the results were complex. I went to trusted institutions and I had second opinions, but even for me, it was hard, and I don't think there's any patient that ever says, are these results equal? And in fact, they're often not. In these times, we need to support science and support innovation more than ever. And that's why we were all here together in Washington and online. We have a large crowd online. I want to thank everyone who's worked with us on this. We have a long road ahead of us, but I think through collaboration, we will get there. To start, I want to join a warm welcome to Friends of Cancer Research President, CEO Jeff Allen, and again, thank all of you who've worked on this.
All right, good morning, everyone. Thank you, Ellen. I was wise and did not wear heels this morning, but you did great. Thanks, everyone who's joining us in the room today, and thank you for the hundreds of people that are joining online for today's meeting. I'd like to welcome you to Friends of Cancer Research's first public meeting of 2025. This year will no doubt be an active period for considering a wide variety of healthcare policies and different priorities, and hopefully it will be accompanied by transformative breakthroughs in the biomedical sciences. Throughout the year, with our wide variety of programs in our portfolio at Friends of Cancer Research, we aim to develop novel data through our research partnerships and help inform future policies with our collaborative working groups. Through venues like these, we'll share the outputs of these projects, engage key stakeholders with the goal of ultimately catalyzing the next generation of breakthroughs. Today's meeting, as Ellen mentioned, will focus on aspects regarding the future of diagnostic testing. It will feature new data from Friends research partnerships, the outputs of ideas from a couple of expert working groups, and we'll cap things off with a discussion of policy considerations for the future. We encourage audience participation in the discussion today, so when it's time, you'll find microphones in the aisles, and we ask that you briefly introduce yourself and by name and organization, and please strive to keep your questions or comments on topic. For those of you who are joining virtually, please use the Q&A function in your Zoom, which will be monitored here in the room so that the questions can be provided to the moderators for each of the panels today. All advanced registrants should have received copies of the white papers and discussion documents that are also available for those of you at your seats and will be available through the chat online as well. I also want to thank all of the participants of our working groups. They've been meeting for months leading up to today's meeting to help develop the material provided. And you'll hear from many of these experts throughout the day today. You'll also notice that many of our collaborators are not able to join today's discussion as the administration transition processes are ironed out. We thank them for their contributions to these projects and look forward to continued collaboration. So without further delay, let's go ahead and get things started. The first session today will feature data from one of our recent research partnerships, the Digital Path Project. This project brings together developers of advanced AI-enabled tools that are able to evaluate relatively routine images to identify specific molecular attributes. The potential for these tools are vast, but like all diagnostic tests, ensuring accuracy and consistency is key. I'm really excited to have the data presented here today. This partnership features truly advanced diagnostic technology. It makes a case for innovative policies and a unique approach to data sharing to ensure accuracy of results. It also shows the power of collaboration to get there. And if we can get all of these things right, these types of approaches hold transformative potential in terms of instigating a new generation of therapies that are just beginning to show remarkable benefits for patients. So to present some of the findings from the Digital Path Project and start off today's discussions, I'm happy to hand things over to Forensic Cancer Research Vice President of Science Policy, Dr. Mark Stewart.
Thank you, Jeff, and thank you everyone for your attendance today. Unfortunately, our statistical team that conducted many of these analyses are unable to participate, and so I'm stepping in to present this data on behalf of our working group that's been contributing to this work, which is our digital pathology harmonization project. This initiative has really been focused on the application of AI-based pathology tools for biomarker assessments. And so today, as Jeff noted, I'll walk through some of our key findings where we've used HER2 scoring as a case study that highlights both the potential of these AI tools, but also some of the challenges and opportunities moving forward. And so as biomarker-driven treatment strategies continue to expand, ensuring the reliability of diagnostic tests, including the availability of AI tools that can help ensure the reproducibility and hopefully reduce variability, become increasingly important. This is especially true in cases like HER2, where emerging HER2-targeted therapies have really heightened the need for consistent and precise biomarker assessments. And so HER2 is a clinically meaningful biomarker in breast cancer. It's often used to determine eligibility for HER2-targeted treatments. And historically, patients that have what's considered HER2 high, which based on ASCO-CAP scoring system is a HER2 3+, typically receive these treatments. However, there are emerging therapies that are now available that have shown to be effective also in patients that have HER2 low, or what's also even considered HER2 ultra-low status, expanding the number of patients that are eligible for this treatment, but also making it increasingly important to have accurate classification to identify patients eligible for these treatments. And so AI-driven pathology models have the potential to reduce variability, improve efficiency, and also could provide more detailed quantitative assessments than what may be feasible through manual scoring. And so, as noted, AI models offer a few potential advantages in HER2 assessment. They can help improve reproducibility by addressing human variability in scoring that we know exists. They can help with scalability in terms of analyzing and sorting through large data sets efficiently. And these models may also provide more granular interpretation of HER2 expression, which could also obviously have clinical implications in terms of identifying patients eligible for these treatments. And while these advantages make AI tools promising for HER2 assessment, it's important to understand the performance of these AI-driven tools, how consistent they are, and the variability that might exist among the tools, but also in relation to pathologist-assigned HER2 scores as well. And so we initiated the Digital Path Harmonization Project, which was designed to evaluate HER2 scoring consistency across AI models and pathologists. And so we were able to leverage a data set that was curated by Dr. Robert Salgado and colleagues in Belgium, which consisted over 1,000 breast cancer whole slide images, which included both HER2, IHC stained slides, as well as H&D slides. These images were then shared with each of the participating tool developers who analyzed these samples and independently applied their models to assess HER2 scores. These data were then shared back with us, and our statistical team ran a series of analyses to understand the types of variability and consistency that existed across these tools and how they related to pathologist scores as well. And so prior to conducting any of our analyses, we established our primary and secondary analyses. And today, what I'm going to present is highlighted here in red, which was focused on evaluating agreement based on ASCO-CAP categorical scoring, as well as examining the level of agreement between the AI models and pathologists. We also conducted a series of analyses trying to understand whether there are certain factors associated with these samples or patients that might also be contributing to any variability that we observe. And so before conducting any analyses, it was important to ensure that the data set that we were leveraging for this project represented patients with breast cancer that one would routinely see in real-world care. The data set that we leverage represents patients that came to a single institution, diagnosed with breast cancer through the course of a year. And so it's important to note that the patient and clinical and tumor characteristics of this data set do closely align with reported data from population-level sources, such as the SEER database. Additionally, on the far right, you'll see the specimen processing characteristics, and because these data came from a single institute, all of that stayed the same. And so that homogeneity really enabled us to isolate sources of variability to the AI models and potentially some of the biological features of the samples. And so with that, we were able to generate some insightful data and findings from this data. And so I'll start first with the HER2 scoring distribution across AI models and pathologists. And what you'll note here is that we had 7 different AI tools represented in this data set, along with 3 pathologists. And so I think what you can immediately appreciate here from this data set is that we see a similar frequency of observations that HER2 3+ categories show a similar distribution across models and pathologists, suggesting alignment in identifying these HER2 positive cases. We note that for the HER2 1+, 2+ categories, we see differences in how frequently models and pathologists assign these scores. And we also note some differences in distribution for HER2 0 cases as well. We next wanted to look at this data at a per sample level. And so here in this tile plot, each column that you see here is an individual sample that was analyzed, and each row represents a different model or pathologist. And so it's easy to go down the column and see what the consistency was and making HER2 scoring for each sample. And again, you can see with this dark blue on the right side of this table that there's less variability across models and pathologists for determining samples that are HER2 3+. However, when you get into these other categories, +1 and +2, we see a bit more variability in terms of some models and pathologists calling them a 1, others calling them a 2, and additionally with the HER2 0 category as well. We next wanted to quantify some of this agreement between models and pathologists, and just looking at the top part of this table, which shows the overall percent agreement, you can see that for categorical calls, there's 65% overall agreement among just the models. And you see a slightly better agreement among just pathologists for categorical calls. However, if you go to the far right where we have the dark blue, you can see that within the models and within the pathologists, you have the greatest agreement, again, in determining these HER2 3+ categories versus HER2 0, 1+, and 2+. And then in the middle here, you see a bit more variability among pathologists and models and distinguishing between a sample that's a 0, 1+ versus a 2+, 3+. And the next is a graph that shows pairwise agreement in HER2 scoring across models. And while it can take a moment to orient yourself to this figure, on the x-axis shows each possible pairing of HER2 scores. The y-axis shows the percent of samples that showed agreement in those categories or disagreement. And what I really want to highlight here is here in these red box plots, which show the percent of disagreement. In a perfect world, these would be at 0%, showing no disagreement. However, we see the greatest disagreement between adjacent scores, and so instances where one model calls a sample a 0 and another model calls it a 1. We also see disagreement where one model calls it a 1+, another model calls it a 2+. And as noted, this can certainly have clinical implications as we're trying to distinguish between patients that have a HER2 0 versus a HER2 1+ in terms of identifying patients that are HER2 low for these new emerging therapies. The next set of data provide some insights into what might be driving some of the variability that we see. And we looked at overall percent agreement and looked at the influence that patient demographics and sample characteristics might have. And while nothing really jumped out as being a major driver, we did note that estrogen receptor status, progesterone receptor status, and Ki67 staining, as noted by the arrows here, do indicate some level of variability. We also note that the sample type, whether it was a biopsy or a resection, may also potentially influence the variability that we see, but it is important to note that this was purely a descriptive exploratory analysis and no formal statistical tests were conducted to determine the significance of these differences. And finally, we did a post-hoc analysis where we conducted a more in-depth review of whole slide images, focusing on cases where AI models and pathologists showed notable discrepancies, and we categorized these into 3 criteria. The first being were cases where one AI model scored a sample a 0, while another scored it a 3+ on the same sample, representing the most extreme discordance. The second criteria was cases where pathologists agreed by at least 2 HER2 categories, and AI models showed discordance. And then the last category was cases where all the AI models agreed And all the pathologists agreed, but they did not agree with each other. And so we had a pathologist systematically re-review these whole slide images and documented the key observations. At the same time, we had the AI tool developers reassess these images in a blinded fashion. They did not know their prior scores that were given, and they did not know which images they were reviewing. And so by analyzing these cases, we were able to identify whether specific histological features, staining artifacts, or other sample types contributed to the variability that we observed. And from this, we were able to note several key factors that contributed to the HER2 variability, such as artifacts and staining inconsistencies that led to discordant classifications, heterogeneous staining patterns in some of the samples that were leveraged, certainly contributed some of the misclassifications. And importantly, after the review process, we noted that there was improved agreement in cases, particularly where artifacts were addressed by the AI models and by the pathologists. And so this is just a subset of the data that we generated from this project, and look forward to getting additional data out through a publication in the coming months. But in summary, our study demonstrated that AI tools show strong agreement in HER2 3+ cases, but we do note greater variability in distinguishing HER2 1+ and 2+, as well as 0 to an extent. And the use of a common data set enabled robust, transparent comparisons across AI models, which have certainly helped identify key sources of variability. And so looking ahead, I think there's certainly an opportunity to think about how common data sets or reference data sets could play an important role in providing transparent evaluation of these tools, helping align methodologies, and identifying sources of variability. And so next, we do plan to use these findings to propose best practices for AI tool development and validation, as well as explore how reference data sets can contribute to this. And so lastly, I certainly want to thank all of our collaborators, the statistical team that conducted the analyses, the team that provided the data set, and colleagues at Forensic Cancer Research for their contributions, and my sincere appreciation to everyone involved in this effort. At this time, I'd like to invite the panel 1 panelists to make their way to the stage, who will then provide additional commentary to the data that I just presented, and more importantly, discuss some of the implications. So I'm going to pass it to Sarah Hersey, who leads Precision Medicine, Translational Sciences, and Bioanalysis teams at BMS, to moderate our panel discussion. Thank you.
Great. Thank you so much, Mark, for the excellent presentation. I think it's quite wonderful to see what can be achieved when industry, academia, health authorities all come together to collaborate. And I'm really looking forward to delving more into the topic and the implications of this with our esteemed panel. But first, I just would like to thank Forensic Cancer Research for hosting us all here today and for the great work that they do each and every day. So with that, I'd like to introduce our panel members to you. Right to my left is Santosh Balasubramanian, who is the Director of AI Products for Path AI, focusing on AI-driven solutions in pathology. Sitting next to him is Mark Gustavsson, who is the Senior Director and Head of Computational Pathology and Strategic Partnerships at AstraZeneca. Next to him is Joe Lenrez, who is the Chief Medical Officer at Boston Gene, with expertise in integrating advanced diagnostics into clinical practice. And next but not least is Joan Mancuso, who is a patient advocate who is quite passionate about emphasizing the importance of accurate diagnostics based on her own personal experiences. And so with that, I'd like us to get started with our first question. So I think we can all appreciate we've seen a rise in the use of AI models in biomarker assessments. And I think what we've seen with the presentation that Mark so nicely provided us is that both AI and pathologists have limitations. And what do you see as the most significant opportunities and challenges that AI presents for improving diagnostic accuracy and reproducibility? And so maybe if we start off with you, Santosh, that would be great.
Yeah. First, thank you to Friends for sort of convening the working group to work on this project. I think for me, the main thing that's exciting is the opportunity to focus not just on what a pathologist is able to do or what AI is able to do, but what kind of the pathologist AI team is able to do. And that's just because pathology is intrinsically hard. And as we, as a field, think about how to implement AI and computational pathology, there are lots of nuances to work through about how to design these models, how to evaluate them, how to deploy them in a way that's safe and effective. And I think one of the biggest challenges is there's so much excitement that there are lots of different, companies, researchers, regulators who are interested in sort of how to solve these problems. And I think it's important to make sure that we're not sort of reinventing the wheel, that every individual isn't trying to solve these difficult problems on their own, which is part of why I think projects like this are so important.
It's great. Maybe Mark, we'll go to you next.
And I couldn't agree more. The project in terms of developing tools for pathologists that are consistent across the board so you can get precise and reproducible answers. And I think, that's the challenge, but I think the opportunity that we have with computational pathology and looking into the future is the opportunity to leverage computational pathology as a tool for pathologists to be more accurate and more precise, but also to be able to identify unique patient populations, doing things that necessarily the human eye can't do, and offers us the opportunity to be more sensitive when we think about HER2, when we think about HER2 ultra low and low, but also as we approaching a world where we're going to have multiple approved ADCs, being able to use computational pathology to not only be more precise, but more accurate, and again, I think Mark alluded to this, identify patients, novel patient populations that are going to respond to therapy, and really the ultimate goal there is to identify the right patient for the right drug. And I think computational pathology really affords us that opportunity to take that to the next level of pathology and really revolutionize pathology.
Agree, really exciting, right, what the potential is. And Joe, your thoughts?
Thanks, Sarah. So I believe the opportunity is, of course, there. The AI wave is here. We're, I think, probably riding in the middle of it. What I think the data that Friends has helped to produce with all these different AI developers has shown that it ultimately comes down to the quality and specifically the representativeness of the data. And that may sound a bit like both, like a challenge and an opportunity. But I think the part that I would like to emphasize, despite AI, we're putting discrete diagnostic categories on a biological spectrum of a disease. And whenever you do this, there are certain areas where that spectrum cannot be reflected in discrete groups. So whether AI can solve that or not remains to be determined. But I agree with Santosh and Mark that AI will likely not replace, let's say, a pathologist or diagnostic modality overall, but it will supplement it. And I think that's an incredible opportunity to have, our, let's call it experience plus a validated AI tool to ultimately render a more precise and more useful diagnostic decision.
Great. And Joan, it would be great to get your thoughts on this topic.
Thank you. I want to thank Friends of Cancer Research for how welcoming they are to advocates to be involved in the different projects. I had my own personal experience of being HER2 positive quite a while ago, and I was very happy to find out that this project was taking place in terms of improving accuracy so that patients get the right diagnosis at the right time. Oh, I was diagnosed in 2003 with early stage breast cancer that was HER2 high. I was tested, it was hormone negative, it was 3+. And I had my treatment, which was standard at that time, of adriamycin, Cytoxan, and Taxol. And then afterwards, I just thought I would consult with another hospital. My oncologist didn't really have a problem with that. She was a clinician, but she was also a scientist who had studied breast cancer in the lab. So I went to another institution, And then they determined from the exact same slides that I was actually HER2 borderline. That's what they called it. Those were the words they used, borderline. And so it was kind of amazing to me that it could go from 3+, where there's a lot of agreement, to being just borderline HER2. And I was kind of shocked, but I began to learn then afterwards that this is not particularly uncommon. That, you know, this is looked at by pathologists in the lab, and there could be different sources for potential misinterpretation or correct or incorrect interpretation of the slides. So I was very, again, happy to hear that this project was going on to potentially improve accuracy, either to be used in the lab all the time or to be used to augment, you know, pathologist results.
Now, Joan, that had to be very concerning as a patient, right? Getting that mixed information coming back to you. I'm sure that was quite concerning.
Yes, I mean, HER2 positive is an aggressive diagnosis to begin with. And I had 7 positive lymph nodes. And I did subsequently develop metastatic breast cancer in 2007. So yes, but I had a good oncologist who was really following me.
That's fantastic. So moving to the next, with the DigPath project findings, what were some of your biggest takeaways? And how do you see these findings influencing future work in computational pathology? Joe, do you want to start us off?
Sure. I think the findings have shown that, let's say, HER2 3+, you know, the aggressive disease, we're fairly good in, let's say, diagnosing that. Similarly, the HER2 0 is a category that I think most of the algorithms showed that it's, you know, working. The other categories, not so much. Why? Well, first of all, over the last, you know, probably 20 years, we are focused on identifying 3+ versus the others. And what you mentioned, this term borderline, meant it wasn't clearly 3+, it wasn't clearly negative, so that wastebasket category of borderline was created to say that needs further workup. So I think in terms of moving forward, one element that I see is the, let's call it emphasis on, you know, these additional categories, you know, with HER2 low being one of them that now has actionable treatment or therapeutic actionability, to really delineate what those categories mean by leveraging what we've always done, meaning you do one test and then you do an orthogonal method. That could be another antibody, it could be another technology like fluorescent in situ hybridization or next-gen sequencing, but we try to tackle the biological problem using different methods. Now, whether those methods then result in data that can be also analyzed with AI remains to be determined. But for each of these additional data, the value of supplementing these critical or, you know, what at first glance looks a little inhomogeneous or non-consensus categories with additional data, plus minus AI, I think that's what the future will hold. So AI cannot replace that by itself, but these additional technologies will help us overcome that problem.
That's fantastic. I look forward to the future. So what additional questions arose from the study for Santosh? Maybe we start with you, because I think the data for many of us was quite interesting, and we had a lot of great dialogue amongst ourselves. So I would love for you to share some of your thoughts in that space.
Yeah, I think one of the main takeaways that Mark highlighted in his presentation was that there is absolutely variability across AI models from different vendors, and just the fact of using AI doesn't necessarily eliminate that problem. I think one of the points that was interesting is that variability between AI models is very similar to the types of variability that we see between different between pathologists. Consistency at the 3+, more variability in the 1+ and 2+. I think additional areas where it would be great to continue the work are, one, looking at the pathologist AI team. We didn't, in this study, include any sort of joint review from both the pathologist and the AI. It was all just AI alone or pathologist alone. I think there's an opportunity to sort of include sort of considerations for how that would change the picture. And I'm also excited to continue working on sort of best practices around how to define reference data sets, how to actually analyze those reference data sets, and even provide recommendations back to to AI vendors about how to potentially minimize this type of variability that we observed in the future.
And Mark, it looks like you might have something to add to that as well.
This is why this project is so valuable, to identify those areas where, to inform model developers how best to perform a model and how to develop a model where you are going to get consistent results, especially with identification of tumor, which is pretty standard, and how to score established biomarkers. So I think this is why this project is so valuable, so we can begin to understand what those variabilities are.
I couldn't agree more. And Mark, you kind of alluded to the next topic I'd like to move towards, because I think as we started seeing these results come to fruition, a lot of us thought about highlighted the need to have really a reference standard set, right? And how could those reference standard sets help us evaluate digital and computational pathology models? And so, as many of you recall, we had numerous discussions. I think, Joe, you and I had some discussions at ASCO. There were a lot of great group dialogue around it. And so one of the things that came up was, there's a lot of heterogeneity in the intended use of the models. And so what challenges could the reference data set help us to address? And how might it actually help us improve evaluation of AI models moving forward? And maybe if we start with Joe, do you want to take it first?
Sure, happy to. So I think worded slightly differently is what are the variabilities that we face when doing IHC, right? For those in the room who know, there's a biopsy, there's temperature, there's storage, there's transport conditions. Some go in the fridge, some don't. Some lay in buffered formaldehyde, some not. It is embedded, it is processed, it's dehydrated, it's putting in wax, it's sectioned at different thickness, it's put on different slides. It is completely user interoperable, and then at some point, a subsection will be stained using a different antibody cocktail, different antigen retrievals, and then ultimately a cover slip, and then you have to scan it to actually get to the point where you can do this. What I just rattled off is happening thousands of times in every histopathology laboratory in the US, and this is by no means fully controlled. It is basically experience-driven, and there's control measures for every single step. But if you envision a data set that could incorporate some of these variabilities to assess, for example, multiple scanners, where a slide set, not like the one that we used in this study, but it could be scanned on 6 different scanners to assess for that variable factor. So on the one hand, that sounds a little perplexing, but at the same time, AI has the incredible opportunity that with some of these data available, you can create what's called embeddings that reflect some of these variabilities. So ultimately, you can create synthetic data sets, et cetera, to emulate this, but that enables things like enrichment for certain variabilities that you may encounter in clinical practice that may not be easily available for any given lab. So, as we move AI into clinical practice, accounting for some of these heterogeneity will come down to the quality of the data, and thereby I think diagnostic tools like the one we assessed here in a group of let's say, developers, can become more accurate and thereby improve the diagnostic quality.
So do you envision that you could almost make minimum requirements to achieve accurate results, that it would help inform what parameters are needed for successful interpretation? Because as you mentioned, there's a tremendous amount of variability. My assumption would be not all algorithms would be able to tolerate that said variability, and that this would be a tool to allow developers to understand how much variability the model could actually absorb to still produce accurate and reliable results. Is that kind of one of the things you would envision?
Absolutely. In other industries, that's actually common practice. I'm sure you all have seen, crash tests. They're not done to test anything else but to see what happens if the system breaks. And in diagnostics, that is usually done during validation. Those data are usually not shared because people, I think, feel that that's sensitive. But having reference standards and reference data sets that truly assess where and when the model breaks is sort of that extreme. I haven't seen a lot of that, at least in medicine, but in other subspecialties, that's commonly done. The other part about the quantification itself, if you ask a pathologist, give me the percentage of, let's call it, your sureness that this is cancer. Are you 97% sure or 77% sure? That is not easy to be done. But with reference data sets, that might be a way to express probabilities. So I think there's great potential related to these reference data sets.
Super exciting. And maybe Santosh, would you like to add anything?
Yeah, I think the opportunity to standardize the way that we construct these reference data sets is really exciting and very important. I think we also need to balance that with the reality that building these data sets is very difficult, and the number of unique use cases for these AI models is vast. And so I think kind of as a field or as a community, we have to collectively decide where to allocate scarce resources in order to build these reference data sets. Because if we try to build a reference data set for every possible use case, I think we'll set ourselves up to fail. And I think there are many cases, for example, that there may not actually be that strong of a sort of a demand or sort of a motivation for people to build like an AI model for a particular use case. For example, if, I don't know, there's a particular type of assessment that's very easy, that pathologists are already very highly consistent at, there may not actually be a need to build kind of reference data sets because there's not a huge demand for us to build AI in that specific scenario. So I think the main sort of challenge for us is to try to think through these cost-benefit analyses and make sure that we don't end up building a large, robust reference data set that doesn't actually get used.
Great. And then Joan or anyone else, would you have anything else you'd like to add?
I think it was important for me to look at patient characteristics in terms of the data set. Things like age, sex, menopausal status, and also about potentially ethnic and racial background. This has become sort of an issue lately about knowing whether this information has an impact on results. So Dr. Melissa Davis of Morehouse School of Medicine gave a plenary lecture at the San Antonio Breast Cancer Symposium where she discussed this, how after 10 years of hormone therapy, that women who were Black did not really have the best results that, let's say, white women. Another thing this was based on was the fact that Black women are twice as likely to have triple-negative breast cancer. And that was studied in part by others, but by Rachel Martini, who went back and looked at African-associated gene expression by looking at African Americans as well as Africans in sub-Saharan Africa, East and West Sub-Saharan Africa, and find out that they also actually have a higher probability of having triple-negative breast cancer, and determine some genes that could be involved with immunologic profiles. It could also potentially depend on what kind of a grade side effect you get. For example, a study that was published in the American Journal of Clinical Oncology in 2021, it was based on a study from Memorial Sloan Kettering Cancer Center, noted that Black women had twice the risk of having heart problems or cardiac problems from drugs like trastuzumab, and that even if it's adjusted for stuff like high blood pressure and diabetes and BMI. So I was kind of very interested as to whether any of this information could be collected, but it turned out that in Belgium, where the samples came from, that they're not allowed to collect this information. However, I'm glad that this would be, and this is true for other countries, they either are allowed to or not allowed to. And of course, this also relates to Asian populations as well. And so, so I'm glad that this at least got on the list of a reference set of data that could potentially be important to know. So I was kind of wondering whether if that data would have been available, whether it would have shown anything in terms of the results. But unfortunately, it was not forthcoming.
Yeah, it is unfortunate that we didn't have that information available, but very, very good points. So both in the Digital Path project and the discussion document describing the potential uses of reference data, there was a push-pull. We all had a lot of discussion about how do you define ground truth or define reference standard to assess the accuracy of the model. So what are we assessing against was a hotly debated topic. And so from your perspectives, why is this difficult to define? And what are some of the considerations when building this reference data set that we'll have to take into consideration? And maybe if we start with Mark, would you like to start us off?
Sure, and it's definitely a challenge. And I think there's two levels for a reference data set. First of all these models rely on the ability to identify tumor. And we think about breast cancer with HER2, you have the challenge of differentiating DCAS from tumor from normal. And I think for a reference data set for all these models, that's the foundation. So if we build a well-represented data set that represents all the multiple histologies across the range, multiple clinical characteristics where model developers model developers can, and it's not just against HER2, it's just, are we identifying tumor? So if you have a well-annotated reference data set, so the model developers know that we're consistently identifying tumor in breast cancer, and all these models are very consistent in that level of identification, then the next level of that for reference data set is obviously the biomarker. How are you calling the biomarker and things like that? And I do think that's where it gets a little contested, where, you know, how do we define what is +1, 1+, 2+, and those types of things. But at the very baseline level, I think a reference data set that can really tell us, are these models identifying the same areas, are really important.
Great point. So maybe we'll pass it over to Joe next.
Sure. I think we just heard some of the population level heterogeneity, right? You know, you mentioned some of the population characteristics that we know track with certain state of the disease. And as Mark just outlined, within the tumor, even identifying the tumor might show some heterogeneity. One element that I believe is also very critical for reference data set is within a human, the heterogeneity across multiple sites. Let me just quickly expand. So when the tumor develops, some cells might actually express higher level HER2, while others do not. That might actually convey a proliferative advantage of one of these cells that has this overexpression. Thereby, that subclone might seed to the lymph node or might seed, let's say, to the brain. Now, when we clinically diagnose cancer, we have to take a biopsy, a tissue sample from one of these sites. Now, multi-regional sampling is usually done after the diagnosis when considered that the extent of disease is resectable. If it's systemic, or if it's, for example, metastasis in the brain where you can't easily get to, we don't even sample that. Now, from other cancers, it's pretty clear that HER2 is enriched, for example, in central nervous system metastasis. So whether a specific tumor sample is truly reflective of the total state of, let's call it the HER2 state of the disease, remains to be determined. So I see, especially when we do extended resection, we heard you had 7 positive lymph nodes. I presume there were some that were negative. That extent of, let's call it anatomic distribution, and how these different tumor cells relate to one another, I believe is providing a much deeper insight into the biology of the disease. So I think that would be, in my opinion, a very actionable element to expand in a cohort or in a reference data set.
Very interesting. And Santosh, do you have any thoughts?
Yeah, I think one thing to remind ourselves of is that we sort of simplify the characterization of the patient tissue as 0, 1+, 2+, or 3+. But in order to get to those slide level labels, we're asking a pathologist to evaluate on the order of tens of thousands of cells in a particular image. And that the fact that we are asking a human to sort of evaluate all of those, identify the specific subset that have a particular staining pattern, and then identify whether that's greater or less than a particular threshold, like 10%. It's just a very difficult problem. And I think that the intrinsic difficulty of the task is why we see so much variability. And I think that rather than focusing just on the idea that there is a single truth and we're measuring AI's ability to get to that truth, I think another way to think about it is sort of think about the level of variability between pathologists and ask instead the question, is the AI performing on par with a pathologist in a cohort? So for example, you could ask questions about whether the comparison between AI and another pathologist, whether that accuracy or that concordance is similar to the concordance that you would get between 2 expert pathologists. And I think using that as another analytical tool, especially in these areas where it's very difficult to get to a single, sort of consensus ground truth across a large data set, gives us another kind of tool in the tool chest to be able to understand how well these models perform.
I think that's great. And I think you had mentioned previously as well that, we looked at it in the context of just an AI-driven result versus a pathologist result, and that there may be an opportunity to improve accuracy and reproducibility utilizing AI as a potential tool in the toolbox of pathologists. So I'd be curious to see, does anyone have any thoughts around that aspect?
Yeah, I guess just to build on that, we didn't assess at all whether the AI pathologist team was better than the AI on its own and better than the pathologist on its own, because we're asking pathologists, the AI and the pathologist, to do a very difficult task. There are certain things that the human will be just much better at doing, given that they're experienced, and then certain types of things, like just counting all of the tens of thousands of cells across the slide, that a computer is going to be better at doing. And I think there's sort of another layer to this of designing the pathologist user interface for how they interact with the AI in a way that makes it optimally easy for them to leverage their expertise where they can, but also lean on the power and the capability of these AI models where they can.
Great. And I think, Mark, you have something you might want to add as well.
No, just to build on that, right? I mean, differentiating established biomarkers versus what AI and computational pathology can do in terms of a novel biomarker, right? Obviously, analytically, we want to match the current biomarker, but ultimately, can we use AI and leverage AI to do things that maybe pathologists can't do? So this is where that difference comes, and this is where clinical outcomes data and testing against clinical outcome, and that's the next level, I think, at this point. Right now, we're kind of sitting at the analytical level, and we've seen the analytical comparison. How do we make it better? Obviously, we're going to need to test this against clinical outcome and things like that, and that's really where we're going into the novel biomarker aspect of things with AI and computational pathology and the vision of what we're doing there.
No, great. So after this next question, we'll be opening it up to Q&A for the audience. So I'd like to ask all of you, if you do have a question, to please move forward to take the mic after this next question. So what do all of you see as the immediate hurdles, whether they be technical, regulatory, operational, needed to leap over, and I know there's a lot, so this could be a long question, to leap over being able to fully realize the promise and the opportunity of computational pathology and AI machine learning for biomarker assessment? And maybe we'll start this time with Joan on what she sees as some of the immediate hurdles.
To me, it had a lot to do with every patient getting an opportunity to get an accurate diagnosis. And sometimes that can depend on other factors, of which some that I mentioned. And that it's important for scientists to understand that drugs could be developed which are not beneficial to everyone, and also assays can be developed that do not represent everyone. So I think that that's like a hurdle even beyond sort of beyond the basic science of developing this particular assay.
It's a really good point. And then, Joe, what are your thoughts of some of the key hurdles that we have to jump over?
And we only have a few minutes, right?
I know, I said this was a loaded question. I mean, from all the discussions we've had, we could probably talk for days on the matter.
So technical, operational, regulatory, yes, yes, yes. I mean, if you think about what I just said about the routine diagnostic workflow that needs to be accounted for, technical, operational, regulatory. But the amount of work that went into the design of these different AI models, we haven't even started touching. So if someone says, let's build a HER2 classifier, okay, that sounds great, and then you can get a result. But the hours and hours of work to identifying the encoder, the visual transformer, the architecture, the code set, are we outlining tumor? Are we segmenting tumor? Are we using zero-shot learning? Do we do a subset? What is our ground truth? What was the scanner used? You know, all these factors we have basically almost ignored. I mean, we captured that post-hoc, but we effectively used a data set, in this case from Belgium, which was definitely not available to anyone during training. So you could call that an out-of-domain data set. And we intentionally used that to check how good the models perform. So that was effectively intentionally taking all the variability out and checking how good are the models. So to me, it was very impressive that the HER2 3+ was robustly identified across these models, which is quite incredible because this was truly a data set that has never been seen by any of the models. And all these variabilities for 3+, the model still performed robustly and concordant across these different developers. From a regulatory perspective, which I believe might represent a hurdle to many, we don't have a data set yet that is regulatory grade. This data set would certainly not pass sort of the litmus test of a regulatory agency because it was only from one side. It does not necessarily reflect the full spectrum of, let's say, population characteristics, et cetera, et cetera. Now, operationally, I think I probably speak to all the different developers. This is not easy to take 1,000 slides, ship them around, feed them through a model. So that's an operational challenge as well. I don't think many labs are equipped to just take in 1,000 slides and push them through. But at the same time, this operationally gives us an opportunity that is unknown. I do not know of a single pathologist who could review 1,000 slides a day. And most of these models pushed it through in, a few minutes to maybe 35 minutes or so. But that is, I think just some of the touch points on the 3 dimensions you raised.
Great, thank you. And Mark, do you have any additional thoughts?
Yeah, just I think from the drug development perspective, right, how do we integrate? How do we get this into a workflow and not from all this is retrospective. How do we get this into a prospective workflow? How do we actually take computational pathology and diagnose patients in real time in a prospective way? And what are the regulatory hurdles, just the general requirements for doing this in a CAP-CLIA lab for a clinical trial? All of these things are things that we all have to navigate because ultimately, right, we want to see this as a companion diagnostic. We want to use these as something that the field will have to use in order to diagnose a patient to give a treatment. So those, I think, are some of the big challenges facing us is just how do we implement this into the clinical workflow, just from a clinical trial perspective.
Fully agree, and I think also into the clinic in the long run, right? I think the goal of developing these tools and getting more accurate and reliable tools in our toolbox is actually having them be used by the broader masses, and that, I think, it should not be underestimated what that challenge might look like. And then, Santosh, do you have anything you'd like to close us out with regards to this question?
Yeah, just to build on that, I think the currently available benchmarks are, maybe 10% of labs around the world have sort of the infrastructure to perform digital pathology, meaning they have the scanners that cost multiple hundreds of thousands of dollars. They have staff that can actually perform the scanning. They have the cloud infrastructure to actually store all of the data that they're generating from these. And I think over the next 5 to 10 years, we'll hopefully see most of these labs start to either adopt or experiment or actually fully deploy the digital infrastructure. And I think the major sort of challenges or opportunities are just making sure that everyone feels comfortable in that journey along the way. We're not going to, even if we had the perfect algorithm, so to speak, we're not going to flip a switch and have everyone be using it the next day. It takes time from an operational perspective, but also just from a human comfort perspective for people to get to the point where we're okay using these and knowing that they're sort of impacting sort of patient treatment decisions.
No, I think it's great. I think, precision medicine is an example in its own right? It's been around for the last 30 years or so, and yet we still struggle ultimately to implement it and ensure that everyone is getting that testing done at the time of diagnosis. So I think it's a good case study, but I think the very positive thing is we've got groups like Forensic Cancer Research and others that really bring everybody together to collaborate, to drive it forward with faster speed so we can get it to hopefully everyone sooner. So with that, I'd like to open it up to the floor for Q&A. Please state your name and affiliation before asking your question. It looks like we have one here on the left-hand side.
Thank you. Really helpful discussion, and also thanks for sharing the insights. This is Cheng Cui from AstraZeneca. I just have a question regarding the ground truthing. Since ground truthing is very important to assess the performance of the algorithm, and also there may be a hurdle just given the discrepancy and also the variety we observed from the study. even for the human pathologists to make interpretation, there's significant variability observed. So normally, as you know, FDA requires the use of adjudication panel to do the ground truthing by using 3 pathologists, and the majority rule may apply. And then just from your experience and also from this study, can you share some insights to see whether you consider the current adjudication panel approach is appropriate, or there potentially be some improvement can be done to yield better results from the ground truthing? Thank you.
Anyone would like to take that one?
I can start. I'm sure there's many thoughts here. So it's absolutely true that there is no perfect ground truth. And this data set was not regulatory grade, like I just mentioned. But there's a few pointers just to get towards that. So I think what this study has demonstrated is the amount and level of complexity related to the data needed to even assess the performance. We haven't even started touching upon the building of the models. That part was basically excluded in this performance comparison study. So in terms of the current regulatory concepts that the FDA has put forth, they distinguish between train, tune, and then test. They use the term test for performance assessment of the final, let's call it, deployable model. However, if you envision that such a model is deployed, it might be deployed in a different setting, so then under CLIA you would have to reassess that performance. Every single one of these steps relies on a ground truth data set, ideally one that hasn't been seen by the model. So like a training, a tuning, a testing, and then an implementation data set. I do not know necessarily how that's even done right now in practice, and I believe that relies 100% on the quality and competence of the local team. That part, I highly doubt that the FDA can regulate that, because that would be considered the practice of medicine under current concepts. Now, to be a little more specific about the quality or the models itself, what I think was revealed in this survey is some of the architecture was assessed. I won't go into the details, I think that's in the paper. But one thing that is known for humans is that if I look at a case, after a washout period of 2 to 3 weeks, I might actually score it somewhat different. So there's an intra-observer variability and an inter-observer variability. In an AI model, that might also be the case, especially when generative AI is used. And some of the technologies underlying that, they're not deterministic, they're probabilistic. Meaning, if you ask the same question to ChatGPT, what that means is you might get 2 different answers, right? And I'm sure you have experienced that. These probabilistic models likely require a completely different assessment of reproducibility or precision for even the same slide. We haven't done this. Would actually be an interesting follow-up experiment to ask people, what happens if you score these 1,000 slides again? And one would wish, similar to a pathologist, that the exact same result is produced. But that is not necessarily the case, because some models might not be deterministic. So what I'm trying to say is the regulatory frameworks are facing currently, even under some of those online learning or generative AI models, probably a little bit of a pressure to adopt fairly quickly. Concepts like predetermined change control plans or agile regulation have been proposed. But what that specifically means and how to account for that is a big issue. Now, until then, I think the approach of having multiple raters, probably from multiple centers, and giving a consensus approach is probably the best we can do, because that is in reality what clinical practice looks like.
Great. We have a question. We'll have a question on the right-hand side we'll take first before going to the online questions. Please state your name and affiliation.
Thank you. Thank you for a very interesting discussion. Janaki Veeraraghavan, I'm from AstraZeneca as well. You touched upon a very interesting concept about deterministic versus probabilistic models and the nature of the variability and outputs over time. And you also said that we have not touched upon model development at all. Can I ask for friends or a consortium to also consider what guidelines we need to put around model development, performance metrics for models, The fact that we recapitulated the inter-observer variability with the models that we see with pathologists pretty much tells me that we trained the models to pathologist performance, right? To an extent, we are recapitulating the same problems that we've encountered throughout, and by deploying homegrown models, we are going to have the same problems that we are envisioning with pathologists. So how would we approach that problem of putting bounds on performance, putting the guardrails that when we talk about accurate results, we build our models to generate accurate results? How would the industry deal with that question? A challenge to friends or other consortiums.
No, it's a great thought. Mark, do you want to?
Well, no, and I think I alluded to what I was saying earlier. At the foundation of all this is these models and how they identify tumor, right? And to what Joan was saying as well, what are the criteria for data sets and training data sets? And I think that fits with the virtual question as well, what that variability is. And the baseline variability for a lot of these models is, what they were trained against. And do they identify tumor in the same way? I mean, tumor is ground truth. I think we can all agree that that is, the same thing that we have to account for. And then moving above that, I don't necessarily have the answer to that in terms of deterministic and things like that. How do we define what a 1+ and things like that? How do we define what those cutoffs are, and how do we maintain that consistency? I think that's a challenge, but I think it's an opportunity that this project can lend to, is the ability to really, much like what I think the FDA put out guidance in 2016 for digital pathology. What are the baseline characteristics and criteria that you have to do in order to get a robust model?
And I think the online question is similar to that fact. They were asking, questions around, the training sets. And I think one of the key things that we mentioned, and Joan kind of alluded to this as well, is the sample type matters. How, Joe listed off a laundry list, how the samples are fixed, the size of the, what scanner was used. There's so many things that can introduce variability. And when training these models, it's actually imperative that you introduce as much variability as possible, including things like ethnic diversity and others. A lot of times models, whether it's, pathology or otherwise, are often trained to a very homogeneous population. And that's really a miss that we have to be much more mindful of as we move forward, is, your training is only as good as the diversity to some extent of the training set that you're utilizing. And Mark or Joe, did you have something else you wanted to add?
Yeah, maybe just a quick historic fact. The idea behind this whole concept was that the original team identified that outcomes differed and it tracked with different intensities of the staining. So they just said, wait, the very positive looking cells had the best responses. So ultimately, what I think to answer that question, ultimately outcome matters. And the fact that we now have HER2-low acting agents, that is basically outcome driven. So I think that is just sort of an important fact to recognize.
Great. And so I think we'll go back to the virtual question and make sure we've adequately addressed it. And then I apologize for those waiting at the mics. I think we'll be out of time and need to move to the break. So the virtual question, which we highlighted a little bit already, was the various drivers of variability were discussed in the presentation. How might this influence variability in how the models were developed and trained? And how might it contribute to the observed variability? I think we kind of highlighted it. But I'd be curious if anyone has anything additional to add. I think it's a very important question that is hard to address. Again, it's something we could probably talk about for a few days. Anyone like to contribute to that?
I guess I'll add, in addition to what's already been said, that the variability matters. It's important to have variability both in your development and your evaluation data sets. And I would say that from a methods perspective, I think I've sometimes heard suggestions, maybe we should just prescribe this is the way that you build models. But the field evolves so quickly that it's difficult to have just a single correct way. And it's important to allow for the field to explore various methods and share and communicate what works most effectively. But I think it's difficult to say this is the one way. And I think ultimately all of these design considerations need to take into account the very specific kind of intended use of the model that you're building. Are you trying to build something to recapitulate what pathologists already do, or are you trying to build a completely brand new biomarker that wouldn't be possible to assess via manual histology? I think all of those are sort of important aspects to consider as we think through kind of the impact of variability on both development and evaluation.
I think it's a really good point with the ADCs and the work being done in that space, that it may be difficult to get ground truth from a pathologist perspective, right? And we have to take that into consideration as we think about what to do and how do we validate these newer models that are coming into play. Anyone else have anything they'd like to add to that one?
I mean, maybe just one quick pointer for drug developers. One variability that you can take out, at least in the deterministic models, is the variability of interpretation, but you have to then really make sure that your model functions like you intended it to use. But if you have that model, that's extremely valuable because you can test across large domain data sets or large cohorts relatively quickly and reliably and hopefully reproducible. So that is a huge potential, especially for looking at trial data or retrospective, review, indication expansion, etc. So I think in that area, not being, or not having to rely on a pathologist or having a pathologist plus a model, that is, I think, increasing the quality of a decision related to, pursuing something or not.
And just real quick to add to that, what is the role of the pathologist in the model, right? In the review, can they negatively annotate, positively annotate? All of these things, I think, lend to that variability as well. And we have to kind of take that into consideration as well. Because it will always be a tool for pathologists. And what's the pathologist interface with the model as well is can lend to that variability.
Great, and with that, I think we'll need to close out. I just would like to thank our esteemed panelists today. I'd like to again thank Friends of Cancer Research for sponsoring the project and the program. I think many of us had a really wonderful time getting to learn and collaborate with one another to try to advance the space. So with that, I'll turn it over to Jeff.
Thank you. Thank you all very much for a terrific discussion, and I really, again, want to provide many thanks to the entire working group that has participated in the Digital Pathology Harmonization Project. We look forward to the forthcoming publication for these data and leveraging these findings to inform future policy considerations. Moving forward, as we think about potential applications and the approach of using a common set of reference data that was discussed during the panel today, and thinking about how they can help demonstrate the performance of new technologies, I'm really excited to highlight an emerging research partnership that's building on these experiences. It's called AI RECIST. As many of you know, RECIST is a standardized set of guidelines used to evaluate response of solid tumors to cancer treatments. It's really been the underpinning of treatment assessment of solid tumors for decades. Similar to the digital pathology field, the emergence of AI-enabled technologies may help augment and further inform the determinations made by medical experts, in this case radiologists. This may help alleviate the potential bias and reduce subjectivity that comes with evaluating tumor images. But again, accuracy and consistency to these new tools are paramount. The AI RECIST will aim first to leverage a common set of images for different tool developers to evaluate so we can understand the consistency in their measurements. And second, to explore additional measurements of tumor burden that may provide additional information important to determining tumor response. We look forward to sharing more details in the coming weeks and months about this emerging project and the work to come. But first, as we think about the next steps for this conference, we're going to have a 15-minute break. So please reconvene around 11:30 back in the room here for our next panel, which will provide insights into validation strategies for rare biomarker tests. Thank you. session. As you're taking a seat, maybe I'll use the opportunity just to highlight our recently distributed scientific report. As hopefully you've had the chance to see in the distribution of this report, it's an aggregate of all of our publications, research reports, and white papers from 2024. I really want to thank all of the 400 different working group members who took place in our various projects throughout last year. Your support is really important and helpful as we advance our mission, and certainly greatly appreciated. If you haven't had the chance to look at it, I would invite you to do so either through the chat where you can find it for those of you online, or the QR code that is displayed on the slide here. We certainly hope that this will be a resource for future drug development, regulation, and policy moving forward. So now, without further delay, we'll get started with session 2, validating diagnostic tests for rare biomarkers. This session builds on previous Friends of Cancer Research white papers that proposed approaches for co-development of drugs and companion diagnostics targeting rare biomarkers. The working group today, however, several of whom were part of that preceding paper, will build on that work to explore opportunities for regulatory flexibility, alternative data sources, and for diagnostic validation, and a proposed tool to support sponsor and regulator interactions. Now, to get us started, I'd like to turn things over to Megan Doyle from Eli Lilly and Company to help summarize the work group's recommendations and kick off this next discussion. Megan?
Thanks so much, Jeff, and thanks to Friends, obviously, for hosting this meeting, but also for hosting this work. So on behalf of the Rare Biomarker Work Group, I'm happy to share with you an overview of the white paper that Friends published last week on innovative processes for validating diagnostic tests for rare biomarkers and indications. So by way of background, I think everyone in this room and probably on this call knows companion diagnostics are often co-developed in parallel with a drug, a targeted therapy, usually in oncology, to identify the right patients for the drug. And obviously, this co-development requires a lot of coordination between the drug sponsor, the device sponsor, the regulatory authority. And this graphic just sort of shows you the general steps in the process. But I will note that obviously this is an idealized scenario. In many cases, these timelines don't align this cleanly. You may be doing the analytical validation of the diagnostic in parallel with phase 1 and 2, while you're trying to get the diagnostic ready to enroll your pivotal study. Or in some cases, you may be enrolling your pivotal study on local testing because that might be more patient-centric, or the diagnostic is not ready for prime time. So it's just important to note that while all these activities have to be done, sometimes they have to be done on a relatively compressed time frame. And that's particularly true when you have a drug that maybe is on a breakthrough therapy designation pathway, or is trying to address a rare unmet need scenario. But the thing I wanted to point out about this slide is that for rare biomarkers or indications, all of this work still has to be done to demonstrate that the device is safe and effective and have it approved by, in this case, the FDA. But we often face a critical lack of samples to conduct the validation steps on this graphic in these rare biomarker or indication scenarios. And that can make the development of the test really challenging. So what did we try to do in the rare biomarkers work group? Well, we really had 3 main objectives. We tried to identify situations where regulatory flexibilities might be appropriate to help enable this test validation and get these tests developed for these rare biomarkers and scenarios. We also tried to, given the rarity of the samples, identify approaches and places where you could leverage alternative sample types or alternative data sources to help validate the test. And finally, we tried to outline a framework for how do you share with the agency, for example, your thinking on this, your test validation strategy, what you are thinking would be a reasonable approach to addressing the rarity of the samples, or where would you want to use alternative sample types or data sources, and what's your justification for doing that, and how can we share that with the agency and have productive conversations between all these parties that need to collaborate on this strategy. So how rare is rare? That's really challenging. Obviously, we had a lot of conversation about this in the working group, and we'll talk about this, I think, in the panel. Ultimately, where we landed was to try to really just define some examples of where we know it would be challenging to obtain the samples you need to conduct the validation of the test. So we landed at an estimated biomarker prevalence of 1% or lower in the population of patients with that specific cancer type, or in the overall population of patients with cancer in the US, is the rare cancer type, does it have an estimated total prevalence of 1% or lower? And I should note, we'll talk about this later, I don't think the goal was to identify a strict threshold, but really to identify places where we know there's going to be a challenge in having enough samples to conduct the validation. So what is companion diagnostic validation? So when you have to demonstrate the safety and efficacy of the test, the data that you generate often fall into kind of these two camps, analytical validation and clinical validation. The analytic, that's how you demonstrate the performance characteristics of the test, but also that it identifies the right patients for the drug. So on the analytical validation side, where you're really establishing those performance characteristics of the test, you're trying to demonstrate, is it accurate, is it reliable, is it precise? And that could include studies like sensitivity and specificity and accuracy and interfering substances. But those types of studies, to complete them and assess and determine the performance characteristics of the test, don't necessarily require clinical outcomes data to complete the tests. On the other hand, on the clinical validation side, you're really trying to show, does the test identify the right patients, the patients who will respond to the drug, the patients in whom the drug will be safe and effective? And as you can imagine, that does require clinical outcomes data. So I think it's fair to say that all of us in the working group would argue that when you need to and where you can in a rare biomarker scenario, you should try to consider alternative sample sources for these validation studies for both types. But there may be clear examples, for example, on the analytical validation side, where you may not need your clinical trial samples to do the analytical validation, and that might not be a good use of your precious clinical trial samples. So what are some alternative data sources or alternative sample types you could use when you're in this scenario? As a working group, we went through and tried to elucidate, in what kinds of situations might it be appropriate to use an alternative sample type or an alternative data source, and what might work for which scenario, and how do we think about and prioritize the use of those precious clinical trial samples. So for example, for clinical trial samples, especially from your pivotal trial, we would argue that you should really prioritize that for the clinical validation where it's really necessary, or for bridging studies. If you've enrolled your studies on local tests and you need to bridge to the companion diagnostic to show that the companion diagnostic identified the same patients, that would be a good way to prioritize the use of those samples. On the analytical validation side, there may be alternate sample types you could use. So for example, you might be able to use samples from a different cancer type, or even maybe samples with a different tissue type where you could be analyzing the same biomarker of interest from those samples. So this is just kind of a highlight of one of the tables we have in the paper, but we go into the paper a little bit more into detail of sort of what are the pros and cons of using these different sample types for these different studies. So then, how do we talk about this with the agency? How do we align on a strategy, particularly where we have to use alternative sample types or alternative data sources? One of the things we did in the working group and in the white paper was produce a snapshot tool that sponsors could use to essentially describe their strategy, their validation strategy. Here's where we think we might use this sample type for this validation study, and here's our justification for why. Or here's our justification for why we think we're in a rare biomarker or rare indication scenario, and we need the regulator to work with us and provide some flexibility in how we would do these studies. So we provided this snapshot basically to enable those kinds of conversations, whether it's at the pre-submission or even to provide clarity in the ultimate submission of what was done where and why. So some key takeaways, I'm sure the panel will discuss their individual takeaways, but a few from the paper that we can share is that regulatory flexibilities really can help here in terms of ensuring that we can generate data to validate the test and support the risk-benefit profile for these rare biomarker and indication scenarios. Additionally, that alternative evidence sources can be utilized for analytical and clinical validation for companion diagnostics when we really have a rarity of samples or a limited availability of samples. But again, sponsors should also provide an explanation for why they think these sample types are appropriate or this data source is appropriate, and share that with the FDA to have productive conversations and align on the strategy. And sponsors can consider using the snapshot tool as a way to have those conversations. So with that, I'd like to invite my fellow panelists from the working group up to the stage, and we'll have a little discussion.
All right, well, thank you, Megan, for doing an excellent job presenting our paper, all of our work, and thanks to Friends of Cancer Research for inviting us here today. I'm Jen Dickey. I'm the head of regulatory for PGDX, now a part of LabCorp Oncology, and I'll be moderating our discussion today. Here to my left, of course, is Megan Doyle, already introduced. She's the Associate Vice President, Assistant General Counsel for Diagnostics at Eli Lilly. Next to her is, of course, the great Liz Mansfield. She's the VP of Regulatory Policy at Foundation Medicine. And then last but certainly not least, we have Elaine Silk. He's the Senior Director of Regulatory Affairs at Tempus AI. I think it's great that we're all here, that we all managed to get through the paper and get it written. I think it's a testament to the persistence of Friends of Cancer Research that they're able to wrangle all us cats and get us to do what we need to do. But I think the outcome has been really great. So in a lot of ways, this paper is like a step two, a continuation of a discussion that we've been having with the agency and with each other for a long time. The first paper was published in 2021. And the 2021 white paper addressed challenges and laid out potential solutions for co-developing therapies and CDXs targeting rare biomarkers. And we noted the need for regulatory flexibilities and alternative validation strategies. So since that paper was published in 2021, what has changed in this time? What in these 4 years has kind of evolved that we kind of went back to the table and wanted to have another round of discussion? Liz?
Thanks, Jen. And let me start by thanking Friends for all their work. I think we've been working together for the last 5 years or so. And shout out to Hillary Andrews, who very happily led our group and made everything really easy. So the difference is what's happened since 2021. I think we've witnessed some amazing strides in cancer drug development that are leading towards more and more biomarker recognition within cancer subtypes and development of drugs to target those smaller populations. So we've had some more experience in more rare biomarkers, and we've seen FDA be somewhat open to using some alternative strategies for validation. And as we move forward, I think we can expect this to happen more and more often. In addition, on the real-world evidence side, which we mentioned in the paper, there has been quite a lot of work and interest in beginning to use real-world data and real-world evidence to support companion diagnostic indications. And I think that we're kind of at the threshold of being able to move into that.
Megan, do you have any thoughts?
Yeah, I would just add, I concur with everything Liz said about having more experience in terms of how to do this and maybe seeing more flexibility on the part of the agency. I think, in my view, the difference between this paper and the prior paper is that we try to get into a little bit more of those alternative data types, alternative data strategies. It's a little bit more practical in terms of not just when do we need flexibilities, but what do those look like and what would be a reasonable and scientific way to approach these challenges.
Yeah, absolutely. Like taking a really pragmatic approach is key to kind of addressing these big problems. So during your presentation, you reviewed our aligned definition of rare biomarkers to be less than 1% of the population with that cancer. So what were some of the considerations that went into establishing that definition, and how sponsors should consider the definition when they're requesting regulatory flexibilities? Elaine?
Yeah, thanks. I think Megan really captured this well in the initial discussion, that this is something we talked about a lot in the group, about how do we define when something is rare. And I think the real consensus was that 1% is not a magic number. Nothing happens when you hit 1.1% or just below that. That's not the determinant for when flexibilities can be applied or not. It's really a starting point for a discussion with the agency on when to apply flexibility and what kind of flexibilities. And I think part of that is probably dependent on the fact that sometimes biomarker prevalence can be really hard to estimate. Literature might have, you look 3 different places, you might see 3 different numbers, and then what's the real prevalence? So, it's not absolute. And then I think the other part is that it's really, in a sense, kind of a proxy for the idea that samples to use in analytical testing would be scarce. And I think there's other factors that come into a consideration for that, including kind of the sample type, so kind of like liquid biopsy would be another factor to consider, and then the technology that's being used. So, this is the 1% prevalence is a good starting point, but there's lots of other things to consider around that.
So, what other characteristics might influence how you consider flexibility? So we talked about, like that 1% prevalence. We argued about the number, kind of fiddled with the wording. But what other types of considerations might influence how you consider when you ask for regulatory flexibilities? So Liz?
Well, obviously the small population size is the thing that we mainly considered, and as Alan said, 1% is kind of a starting point, not magical. But there are certainly trials where the samples that may have been obtained for initial testing are no longer available for many different reasons. They may have been used up. They may have aged out of stability where you could expect to get a reasonable test result from them. There are a lot of sort of technical reasons for why you might not have samples available. And I think the paper, we emphasize a lot in the paper that, it's mostly about rare, but at the end of the day, it's about justifying what you're doing to the agency. So you need to convince the agency, if you're having any kind of problem, that this is realistically a problem that's going to prevent you from doing, sort of the idealized development and validation, and that then you can discuss with the agency what the best alternatives are. But I think there's a bunch of things that can play into it.
Yep. Elaine, do you have thoughts?
Yeah, I think kind of another way to also think about this is that samples are rare, but then kind of within samples there might be factors that influence where to prioritize them and when might be appropriate to exert more flexibility. For example, the idea, and we briefly mentioned this before, that for liquid biopsy, the yield of nucleic acid that might be available from a sample might be low. Or the same might be true for solid tumors where the biopsies are small or the number of cancer cells within a biopsy is low. And that would be a case where, for those sample types, you might think about studies like Precision that typically require a lot of replicates. That would be a place where you could focus discussions about flexibility to talk about kind of sample scarcity, but also like nucleic acid scarcity within individual samples.
Yeah, absolutely. We kind of focused on tissue for the purposes of this paper, but considerations for liquid biopsy have been worked on for many years by groups such as BloodPAC and Friends of Cancer Research for quite a while. So we've kind of discussed it in terms of rare versus not rare, but how do you think about differences and similarities between like the rare and the non-rare biomarker CDX validation? Like, what would be kind of the considerations you would put forth to the agency or in considerations for the design of your validation? Megan?
Yeah, well, I can speak from the therapeutic sponsor perspective. I think we have to balance and juggle a lot of competing priorities in this space. You know, we want to develop accurate and reliable tests, but we also need to enable patient-centric trials and try to develop therapeutics, particularly in these areas of a rare biomarker where there might be high unmet need, you know, as quickly as practicable and get those to patients. So I think we have to juggle things like we may have to do things like enroll on local tests. We might have to go where the patients are because it can be hard to find these patients. And they might have an existing test result. But while it allows patients to get on trial more quickly and it can avoid delays, and that can also maybe be more patient-centric in that patients don't have to get rebiopsied, as Liz mentioned, the tissue might be exhausted because it was used in the clinical care and management of the patient. And it's not necessarily as easy in these scenarios, again, because of the rarity, to find other clinical samples that we could use. So then you might say, okay, well, maybe we mandate an additional biopsy in the protocol, but we have to juggle a lot of different stakeholders who might push back on that. You might have patients and providers and IRBs that don't want you to put a patient through an additional biopsy just to validate a test. And then additionally, you know, our drug development programs are global usually, and we may have to develop multiple companion diagnostics for different geographies depending on what platforms are available in that geography, in the labs there. How do we make this accessible to patients? And all of those regulators will want to see those tests developed and validated and demonstrate safety and effectiveness. So I guess I would say that all those considerations just go into play in that the samples are even more rare in these scenarios. You might have to juggle all of those things in any companion diagnostic co-development program, but when you have a rare biomarker or indication, you're strapped even more so in terms of the availability of the samples.
Absolutely, absolutely. Anyone else have something else to add?
Well, yeah, I would add that we emphasize, I think, throughout the paper that really the clinical validation is the place where you want to use where the patient samples have the most value. And so we focused a lot on things that could be done in analytical validation where outcome isn't really necessary. It's not 100% important that, you know, that the same patients that were in the trial are used for the analytical validation. And I think that prioritizing clinical samples for clinical validation and then sort of trying to work around that in analytical validation was a point that we tried to make very clearly.
Yep, absolutely. So when we were assessing the alternative sample types, we categorized and kind of attempted to establish a hierarchy, right? To Liz's point, like clinical validation is maybe the top of the hierarchy, the thing we are willing to spend clinical samples on. But then we would kind of establish a priority for when to use certain samples. And so, Elaine, maybe could you discuss some considerations that went into our big debate of the categorization of when to use clinicals versus other models?
Yeah, I think that was a tough one because it's not that easy to be very objective about you know, exactly what is the value of the different analytical tests and how many samples should be diverted to this test and how many samples should be diverted to that one. I think the way that we really thought about it was kind of holistically, so that you know, in combination, when you're thinking about what samples you have available, given that some are prioritized for clinical testing, how do you allocate the rest, and how do you assemble other sources of information and data to kind of supplement what it is that you have? I think one good example is the use of cell line materials for this, which, you know, can provide a way to access biomarkers in, you know, defined and well-characterized materials, but in some cases, I think it's going to be important to think about, well, do the cell, when we're thinking about using cell lines for a certain analytical study, how well do they represent the patient samples, and how well do they need to represent the patient samples in order to be used in that study? And sometimes if they don't have the complexity that you would want to have represented by actual patient samples, then can you mimic that by manipulating those cell lines, for example, processing them through the normal embedding, fixation embedding that a patient sample would go through to capture some of that and bring a little bit more robustness to the use of those samples in the testing.
Yeah, absolutely. It's when is DNA, and we can use it from alternative sources, and when does clinical context actually matter? And yeah, it became clear during our discussions that in the sense of clinical context, maybe clinical variability matters, but in some of these analytical situations, we have tools that we can use. So what types of clinical samples could be considered as alternative samples for clinical validation, and how? Because we just said, we're going to use all our clinical samples for clinical validation. But anyone who's ever been through a trial knows, you don't get all of them. You never get all of them. They disappear into the ether, and you're left to struggle through clinical validation on your own. So Megan, maybe from a therapeutic standpoint.
Yeah, I think it's challenging for all the reasons we just discussed. You can have a situation where you want to preserve your clinical pivotal trial samples for clinical validation. You still don't have enough, or they're not available for all the reasons that we've already cited. So I do think that's the more challenging scenario. What do? You might be able to look at samples from earlier phase studies in that same drug development program where it's the same intended use population. That might be an option. Or maybe you can look at how do we structure our pivotal trial to really try to capture the samples and the specimens as much as possible from all of the different participants. But of course, there's challenges there. As we articulated earlier, you might be better off, trying to require archived samples. That might, be more palatable than asking patients to go through another biopsy, for example. Or you could kind of try to think about in advance, are there alternative sample types that we could use if we anticipate being in this scenario because it's a rare biomarker or a rare indication and the trial's already really small maybe for patient numbers. Can we capture, collect blood as well? Is that clinically and scientifically appropriate? Or could we pre-process or bank pre-processed samples, RNA and DNA extractions? Would those be something we could use as an alternative? But I think all that just points to, as the therapeutic sponsor and working in concert with your diagnostic partner, you have to develop a comprehensive biomarker plan and specimen collection plan. And how are we going to educate sites on how to capture samples, how to preserve samples and store them? What is our plan for alternative data sources if we think we need them? And how do we get ahead of this so that you make sure you're not using those samples for exploratory analyses or something when you really need them for the CDX clinical validation? So sponsors really have to think about that up front and develop those plans in a really comprehensive way so they have a strategy for how they're going to do that clinical validation and have, tried to get as many samples as they possibly can.
Yeah. And speaking of alternative data sources, maybe one evolution from 2021 to now is maybe the rise of real-world data. As a concept, though I haven't seen it actually much in practice quite yet. Elaine, do you want to speak to our discussions on real-world data?
Yeah, I think it's something important, and we wanted to capture it in the paper, so we kind of carved out a row for this in one of our tables of alternative data sources. And I think just taking a step back, in terms of talking about how to support a comprehensive package of analytical validation, it's not necessarily all about analytical testing. I think in some cases, if there's going to be a little bit more of a gap in one area, then maybe other types of information like real-world data could come in and support that, and that's really the place that it fits. I think it can be a little harder, though, to think about how real-world evidence could be used for kind of CDX clinical support in the pre-market, because oftentimes you'd be thinking that the test would be used to identify patients in the intended use population who would get the therapy, and that's just not going to be happening really in the pre-market setting. So that's a difficult case. But to whatever extent that data collection around that might be possible, or in kind of parallel situations, then that could really be useful to supplement to supplement the whole package of information that's being provided. I think it is important to remember, though, that real-world evidence is not like clinical trial data, and there can be important differences. Not that it's necessarily better or worse, just that things are measured in the clinical trial in one way, and they're measured in the real world in another way. And when using real-world data, it's just important to recognize those differences and be able to explain to the agency, what they are and the implications they have, and provide a justification for why that's acceptable, given the context that you want to use that information in.
Yep. So moving down from the clinical samples, since we already know we ran out of them in our clinical validation study, our paper, we reviewed samples like our less ideal samples, like procured samples that are unrelated to the clinical trial. We reviewed cell lines. We reviewed these more in the context of analytical validation. So Liz, what do you think are the key considerations for selecting which samples or sample types you would put into that analytical package?
Yeah, so you mentioned a number of alternative possible sample types, and I think one of the underlying things that you have to think about is in a particular type of study, for example, precision or linearity for quantitative assays and so on, is what are you trying to show with that study, and what are the properties or the characteristics of the alternative sample that support that completely. So for some things, like interference, a cell line isn't going to be helpful because the cell line doesn't have the interferences that a human sample might have. And on that is really to think about what am I trying to show here, and what are some alternatives that would actually demonstrate that in a robust way.
Yep, absolutely. And in the white paper, we kind of emphasize the need to kind of have an ongoing discussion with the agency. We talked about how we've got to kind of devise a validation strategy and then defend it to the agency in either the pre-submission process, ideally, right, before you've spent millions of dollars on studies, and in the marketing submission at least, right? And we put tables into the paper to try and help create a tool to enable that discussion with the agency. So Megan, what do you think, what is the theory for using this document? Like, how do we wish people to use this document to help with that kind of maybe back and forth with the agency?
Yeah, I think our intent was, not to create a document that everyone has to use by any means, but something that could help streamline those conversations, provide the information to the agency up front about your strategy and your rationale and your justification so that you can have a productive conversation with the agency, and giving people a tool or a way to maybe categorize their thinking and put it on paper, and really to also help devise that validation strategy, looking at it and think about, do I have a rationale for why I think this is appropriate, and what is that, and how do I document that, and how do I share it with the agency, so that rather than just kind of coming in and saying this is what we're doing, you can actually justify your approach. Because I think Liz said it earlier, and I think that's also an important takeaway, is that it's up to sponsors to justify to the agency their approach and why it's scientifically valid, why it's scientifically appropriate. And I think the tool is just designed to try to give you a vehicle for how you do that.
Excellent. Liz?
Yeah, it probably could serve as a good planning tool for companion diagnostic discussions between the sponsors, the diagnostic and the drug sponsor as well, to actually try to get through this, because, I'm not sure we really emphasize this, but at the end of the day, for these rare biomarker companion diagnostics, the goal is to get a validated companion diagnostic on the market to support that drug. Is it a perfect validation? Is it the level of validation that you'd really like to see if you had, everything fell into place exactly? And the answer is probably not, but you should be doing the very best you can, because patients actually deserve that. They deserve a good test. So I think this is a great planning document, a great discussion document to try to get to the best solution.
Yeah. Elaine?
Yeah, I actually just wanted to add on to the part that you mentioned about patients, Liz, that like patients deserve this, and also their samples are precious, not just because they're, they come from clinical trials and there aren't many of them, but patients have contributed these, and they should be used in the right way to provide the right information. And there's not unlimited samples, and so it's important that we wanted to, what's in the paper reflects to some extent things that are already being done, but maybe just weren't clear to all test developers. And so, this was really one of the motivations to bring this information forward, was just help people understand that there is not a absolute formula for how to go through this. There is already flexibility, and these are some of the ways to think about it.
Absolutely, absolutely. I think as we move forward, and maybe, who knows, in 4 years, we'll be back here with step 3, like, the robots have taken over and now we don't need samples anymore. But until that time, right? So what do you think, Megan, are some of the key takeaways that we want our audience and the readers of the white paper and our discussion here today to take away from the paper?
Yeah, I think some of my key takeaways are just kind of how we were discussing patients. We have to be really thoughtful about what's best for patients as we're doing these co-development programs. How do we get therapies that are safe and effective to patients as soon as practicable? How do we get well-validated tests that are, we feel confident in to patients as soon as possible? But how do we also take good care of their samples and make sure that we are not putting them through unnecessary procedures where we don't have to, if there are other flexibilities that could be applied. But I think the other key takeaways I would say from my perspective are that these scenarios are real, where we do have these rare biomarkers and indications, or for other reasons, as Alan mentioned earlier, we might have challenges getting the samples we need to conduct the validation studies. So these situations are real, and the regulatory flexibilities really are needed and really do help to enable getting a well-validated test developed and out to patients. And then lastly, I would say, I think this just harkens back to what Alan just said, that regulators and sponsors are working on this already and are collaborating really well in these situations. I think what we were just trying to do was set up, a framework for how can we all think about this, how can we all get on the same page and maybe collaborate even more efficiently on this and make some of these things maybe more of a standard practice where it's appropriate and scientifically valid.
Okay. Liz, what are your thoughts?
Yes, thanks, Megan. I agree with what you said. And I'm sorry our federal colleagues could not be here today. The FDA were observers in our working group. And it seems, I can't speak for them, that they felt we were maybe on the right track. They weren't looking on in horror.
They didn't yell at us.
So I think they're open to some of this, if not all of it. But at the end of the day, I'll go back to patients. Patients deserve high-quality tests, especially in this arena where the test is determinative. You're going to get this drug that may prolong or save your life, or you're not going to get this drug. Well, first of all, of course, the test has to be run, but you want the highest quality test you can. And so the goal is to get the best test out there in a timely manner. We don't want to wait for the perfect effectively. At the same time, I think as we propose this, we're doing it in the context of the rare biomarker or the rare cancer. This isn't an attempt to lower the bar and say that it's not important to do the best validation you can for companion diagnostics. It's an attempt to recognize the realities of drug development, companion diagnostic development, and to set out a plan for how you might actually get to having a test that is useful and reliable for patients. So I think, we want to be careful there, and I just want to add in here, I kind of skipped past it, but I think especially in this rare biomarker arena, and you guys can tell me, but it's really important to have a prospective biomarker plan going into the therapeutic clinical trials, especially when the biomarker is going to be rare, because it creates so many issues when samples aren't available. And I would encourage, if it's not being done for the diagnostic side or the clinical side the pharmaceutical and biological drug developers to really consider a prospective plan for how you're going to deal with this.
Absolutely. Elaine?
I mean, I think that the points about these situations being real is kind of one of the most important things that I think it's important to remember, and that there are ways to work around them, that just because samples are scarce doesn't mean that there's no path forward. I think the important thing to keep in mind, though, is that scarcity of samples isn't itself a justification for doing something different. And that's why I think the snapshots tool that we put at the end of the paper can be really a helpful way to try to, you know, for your own purposes as a test developer or in partnership with a therapeutic developer, articulate to yourselves a rationale for doing something different, what that different approach is going to be and why it's acceptable, and then also use that as a tool to go and talk to the agency, because you really need to be able to you know, understand this yourself and then present it. And I was thinking about the idea of these snapshots, and I think that's a really apt way to put it. But then there's also this, you probably have all seen these pictures where they take a bunch of different snapshots and turn them into a bigger picture of something else. And I think that's also a way to think about these snapshots, that each study itself is a little snapshot where you're potentially taking a different approach to providing information to support a certain analytical characteristic, but holistically, the full package of information that you're presenting is also important to consider.
Okay, so as we start to move towards the question-answer session, the stands for the microphones aren't there anymore, but rather if people can raise their hands, then microphone runners will be available to take the microphone to you so that we can begin the question-answer stage as we start to wrap up our discussion. So I see some hands.
Hi, I'm Raymond Perini. I'm from the Milken Institute. You all mentioned the importance of incorporating what patients need and making sure that their needs were taken care of. I was just curious if you could share a little bit more about how you kind of worked with the patients to understand what they need, and also how they may have been engaged or involved with the development of these validation studies.
So I can take that. We didn't have a patient representative in our working group, as far as I'm aware. We were primarily a group of diagnostic developers and pharmaceutical folks who have been in these scenarios before and have seen the issues arise. It would be very interesting, however, to have a patient perspective on this as to, you know, how patients actually feel about perhaps giving a second sample, or the use of their samples anyway. There's a, you know, the informed consent, I'm not sure is always perfect around how samples get taken and ultimately used.
Yeah, and I could just add that, you know, I do think many therapeutic product sponsors try to capture the patient voice or the patient perspective when designing their trials. There's various mechanisms for doing that. And if you look at sort of FDA's patient-focused drug development work, there's a lot of ways in which sponsors think about this and try to incorporate it into how they design the studies. I think it's probably, to Liz's point, maybe less often that people are drilling down into the patient perspective on the validation plan or the test and the sampling plan.
Yeah, that's true. Another question?
Yeah. Hi, this is Sid from Merck. So great topic, work, and discussion today. And I understand that this is just a guide and that there are various factors that play into it. But I'm interested in that 1% definition that you set for rare biomarkers, because like even 2%, 3%, 5% could be challenging to source. So did you think that going beyond 1% would be difficult for the regulators to digest, or where did you how did you land on the 1%?
Yeah, and I would say 1% was a bit of a negotiation with the agency. And to your point, like there are clinical contexts where 5% prevalent biomarker would be very difficult to source, particularly in liquid biopsy instances where you can find the biomarker, but there's not an abundance of cfDNA to do replicates or appropriate analytical studies. So the 1% was a bit of a negotiation, kind of in the how many do you have to screen to obtain one, how many would you have to get ultimately? But Elaine, do you want to speak to it?
Yeah, I mean, I think, it's a good point. And I think it goes back to the idea that 1%, we felt, again, it's like, it's not a magic number. It's not a hard cutoff after which, if the prevalence is 1.5%, there's no room to do anything different than what you would do if you had an unlimited number of samples. And so I really think it comes back down to, explaining why the samples are hard to source, or why there's not enough material available from samples, or why it's difficult to obtain enough samples to conduct kind of, comprehensive analytical characterization that you might do if you had, like, unlimited access to samples. So I think your point is good, that there's no real hard cutoff. And, my impression of the agency participants in our working group was that they were open to this idea, and they also didn't believe that there was kind of a number after which they wouldn't consider further discussion.
Yeah. And I think we did a little back of the envelope calculation as well, as what would 1% be in lung cancer? What would 1% be in an independent cancer? And as Ellen just said, I think a number of these concepts can be carried over to situations where the biomarker isn't necessarily that rare, but other factors are present that result in a really limited ability to obtain appropriate clinical samples. So I would say a lot of it is really a guide to just a discussion with the agency, maybe even if your biomarker isn't rare.
Yeah, and I would just add that I agree with everything that's been said, but that's where the justification is really critical. There may be a really high unmet need. Again, there may be a patient population where not only is it challenging to source the samples, but there's really high unmet need. And the FDA is usually very willing to engage and have these conversations about flexibilities and partner with the companies in those situations.
Absolutely. I've also had the experience where the agency has been flexible about sample numbers, even when it's not at a 1% hard and fast cutoff. So the point is to have the discussion. The point is also to make sure you have a justification for your actions and that you're capturing the needed parameters. I see other question there.
Hi, Kelly Mercier, Daiichi Sankyo. So thank you for the discussion. It's been very interesting. I think we've discussed, one side of the ditch where we exhaust all of our samples in the analytical and clinical validation. The other side of the ditch is that we leave specimens forever in the -80. So how do we encourage sponsors and agency to disseminate that data in some fashion, regardless of the clinical outcome for a therapeutic?
So disseminate the data from the samples that are stuck in the -80? Is that what we're kind of thinking of? So this gets to maybe some real world evidence or additional evidence that we could have. Truthfully, honestly, the thought that there's samples living in a -80 makes me very excited to have the keys to your -80. In the case of rare biomarkers, it's actually extensive and expensive search efforts. So Daiichi might be sitting on an income stream. But I don't know.
Realistically, there are potentially a lot of uses for samples that are sitting in the -80.
Absolutely.
Exploratory studies, assay development, all kinds of things that ideally are not going to exhaust your most valuable samples that are really needed for a specific purpose. So if people are, saving those up for a rainy day or something, I guess that's their choice, but it seems like you'd want to use them to the best purpose possible. I'm not sure the agency, I can't speak for them, but I'm not sure that they can force people to use those. But certainly there are opportunities.
And Liz, I think it's back to what you said before, which is, using samples for the right purpose to generate the information that you need. And at some point, there's just going to be diminishing returns. So just because you have extra samples doesn't mean that the best use is to conduct, to do 10 more samples for analytical accuracy or 8 more samples for precision testing. There's other work that could be used, those samples could be used for, and I think that's an important thing to keep in mind.
Mm-hmm. So we do have a virtual question, which is, did you also consider patient-derived xenografts and or organoid samples, which may come not from clinical trial patients? So I would say we didn't spend a lot of time on xenografts as a model system for contrived samples, mainly due to just the complexity of generating those xenografts, and then what additional, what would that give you, right? Above and beyond maybe cell line derived or patient sourced from other sources. But do you guys have any thoughts on the use of xenografts or other models?
Yeah, I think, these are really interesting models and could be useful, but the key here is how you justify to yourself and to the agency that these represent a sample in a meaningful way in the study that you are doing. Maybe for certain things, they're quite acceptable. Maybe for other things, they don't quite fit because maybe they don't have complexity, or maybe you can't actually validate what's in that organoid. I don't know that much about them. But I think that, probably anything is fair game if you can appropriately justify why it is, scientifically valid to use that particular alternative.
All right. And so do we have other questions? Oh, yes. Sir, I think. Okay, so Jeff is giving me the nod to get off the stage, so I think we'll.
I think we have to wrap up.
I do
we'll go
think,
ahead and adjourn.
yeah, I do think Jeff is saying it's time for us to wrap up our time, but I do thank you to Friends of Cancer Research for your time, and thanks to everyone for a very engaging discussion.
All right. Thank you. Thank you. If you haven't had the chance to read the paper that this group and their colleagues worked on, I would certainly encourage it. I think it will be quite informative to these instances that will continue to be encountered to come. We're now going to take a break for lunch. We will reconvene here in accordance with the agenda at 1 o'clock so that it's predictive for the folks that are tuned in online. Hopefully they'll have a chance to grab lunch as well. For those of you in the room, lunch is just outside these doors in the foyer. You're welcome to mingle, to eat in this room. There'll be service in here as well. So please enjoy, and we'll see you back here at 1 o'clock. pathways that appropriately balance patient safety and innovation. So to start the discussion, I'd like to welcome the panel up to the stage and introduce our moderator today, Rachel Shear, who is a partner at Manette Health. Come on up.
Great. Well, thank you for having me, and thank you, as always, to Friends of Cancer Research for really leading the way and taking on critical projects of critical importance like the one that we're all gathered here today to talk about. So as Jeff mentioned, in this panel, we're going to sort of take it up a level and talk about what are the important policy considerations that everyone should be thinking about as we think through the lessons learned from both of these really important sessions that we had this morning. I think we heard on some of the panels today that there's already a lot of really great collaboration going on now between the regulators, between FDA and the sponsors. And what we wanted to do on this panel is sort of dig a little deeper and think through what are the systems, what are the policies that need to be in place to enable that kind of collaboration and positive work to continue in a more standardized and widespread way. So we have a great panel here today, and I'll introduce everyone. I think all of us here have some combination of FDA regulatory experience, experience in the White House, experience in the administration, and also experience on Capitol Hill. So it's a lot of really great and unique perspectives here, and we're excited to get started. So first of all, immediately to my left is Joe Franklin, who is now special counsel at Covington and Burling. Next is Rohini Kosalu, who is a venture partner at Fusion Fund. And next, Lauren Silvas, who is Senior Vice President of External Affairs at Tempus AI. So I'll get us started with a question that I'd like each of you to respond to. Each of you brings a perspective to this discussion on AI in healthcare. Can you share a bit more about your background and how you are approaching AI regulation, innovation, and policy development in your respective roles? Starting with Joe.
Great. Excellent to be here, and thanks to Friends of Cancer Research for the meeting, but also for the presentation on the digital path, or the AI Path project. And I should say that watching that made me think of my kind of core piece of background, which is that my dad was a pathologist, and I grew up just idolizing his work in the histopath lab, and getting to go in on the weekends and do H&E staining, which probably wasn't the best from a chemical exposure perspective as a little kid. I think to at least my dad's initial dismay, I became a lawyer eventually, and ended up working as a lawyer for a number of years at FDA, and really thinking about the nitty-gritty of FDA regulatory law, and got to spend a little bit of time in the, a few years in the Center for Drugs, working with review teams, kind of at the interface of law and regulatory review. I then had a sojourn in the tech industry and worked for a few years thinking about health tech and the regulatory challenges and kind of strategic challenges posed by emerging regulations in health tech. And I'm back as at a law firm in DC, really thinking about the interface between technology and our medical products regulatory landscape. And I'll just leave you with one thought that guides my perspective on artificial intelligence and regulatory policy, which is that I think we're all talking about a fundamental challenge that the institutions we all work in are just not very good at incorporating intelligence that comes from computers or comes from a kind of non-human entities. And I think that is, if we kind of think about some of the fundamental principles, which hopefully we can do in this wide open panel on the final panel, I think it will help us identify what some of the priority areas are from a policy perspective.
Rohini.
Hi, everyone. So I'm Rohini Kosalu. I spent many years, almost 20, on Capitol Hill, working for particularly Michael Bennett of Colorado, Senator Michael Bennett, who was on the Senate Help Committee and the Senate Finance Committee. So was lucky enough during that time to get to see healthcare legislation from both a regulatory side and the payment side. So when thinking about products, just to be able to see really how something goes from idea to all the way patient access, does that patient get it, how much do they have to pay for it. I worked for Debbie Stabenow, Michael Bennett in the Senate, and then I was chief of staff to Senator Kamala Harris. She then became vice president, as you know, and then I was her domestic policy advisor in the White House. And I was lucky enough to sort of start on Capitol Hill during healthcare reform, and then sort of many FDA reauthorizations through the years, and got to work with a lot of people on this stage on things like breakthrough therapies and so many other pieces of legislation, Cures 2.0. And then when I left the White House, I started an AI fellowship out at Stanford, working on AI research. So a lot of you know Stanford is doing some cutting edge research there, and then also have joined a venture fund as an advisor for health and technology AI in early stage Series A investments.
Lauren. Thanks. I'm Lauren Silvis, Senior Vice President of External Affairs at Tempus AI. I had the good fortune of working on digital health policy issues in the Device Center, and then also with Commissioner Gottlieb. But at Tempus, it's just been incredible to see those issues come to life and the products and services we're developing to offer to patients. So we use AI and think about AI in a number of different ways. First, like internally as a company, we're constantly challenging ourselves to develop agents, and how can we use AI to look at data, to do our jobs better, more efficiently. And so we're just constantly thinking about it as we go through our day-to-day. But then we're also developing a number of products and services for partners and physicians and patients. So, for example, we have care gap algorithms that where we can use AI to help sort through information in a patient's record and help a health system identify patients who may benefit from biomarker testing, for example, based on the latest guidelines and best practices. We also use AI regularly in our development of diagnostics, where we are analyzing the data that they're generating and then developing algorithms to make better predictions for patients as part of our testing services. We also have developed software as a medical device that's purely AI-based and brought that through FDA, so have the traditional medical device framework-based AI. And then we have lots of partnerships where we're using AI to help with drug development, to better identify targets, to stratify patients who may benefit from enrolling in a clinical trial, to identify those patients who may benefit from enrolling in a clinical trial. And so we're sort of deploying it in a number of different ways at the same time that the policy around it is developing.
Great. So as we all know, in FDA's regulation of medical products across the board, there's this constant tension between patient safety on the one hand and allowing innovation to flourish. So I want to ask Rohini and Joe, as you think about AI-driven diagnostic tools, what are sort of the key regulatory challenges that are at play here so that patient safety on the one hand is preserved and innovation can flourish? And also, do you think there are current proposals out there now that strike this balance?
Okay. I think there's some aspects of regulating artificial intelligence or, guardrails that we can look at what already happens in the context of, when we think about diagnostic tools. And some of these conversations, have been going on for many years, which is, what is the risk of information to the patient? How high is it? Is the patient going to make a life-changing decision that they may regret if the tool is, going to work or not work, or if it's inaccurate? So there's always going to be, you know, just the approach of just having more guardrails around when you're going to make something that in healthcare is a much higher risk decision for you and yourself. We could apply some of those frameworks certainly to artificial intelligence, and the FDA has tried to do that in many respects. I think some of the tension points that we're seeing now is, as Joe pointed out, largely with regulatory bodies. You know, there is the world is much more better for them based off of stability, and the world that we're living in now with AI is largely based off of iterative updates, and they can be big updates or small updates, and how we process those and just how the regulatory environment approaches those are something that's going to be really important as we go throughout. Because I mean, I think just even the other day with OpenAI, for example, releasing this deep research and people being able to do scientific drug discoveries that would normally take people years to do. We've seen statistics where we can look at drug discoveries and they can be potentially down the line 50% faster due to AI. Those are all things that we're on the verge of seeing. And so how do we have a regulatory body that also then complements that?
Yeah, I think this is a great, number one, a great group to be talking about this in. I think that I'm eager to hear whether there are questions in this area, because certainly there are a lot more challenges than there are answers. I am optimistic about AI and this AI medical product development, AI in medical products, and AI in the regulatory context. But I'm also filled with anxiety in this area because the pace of change is so high. And I think it's important to acknowledge that there have been for a long time, that there always will be, and there certainly are now fundamental questions about kind of the legitimacy of different types of regulatory approaches. And I think, you know, from a broad societal perspective, the increasing number of, you know, generative AI applications, as FDA has acknowledged, I think in a really interesting meeting a few months ago that focused on the frontier challenges of generative AI. I think to the FDA's credit, they have put a lot of resources into some of these really kind of on the horizon type challenges. The fact is, people are using these same tools in their pockets on phones. If you're using a large language model based query tool, I think there's just a level of dissonance between where the regulatory conversation is and where the tools are for us all in our daily lives. So I'm worried on that kind of frontier level. I do think, though, that that on the FDA side, especially, so much work has gone into guidance development. And one, I guess, just to, you know, there's a lot of things we can talk about here. One thing that I'm very eager to watch, and one I think very promising development, is that because of AI and because of the uses of AI, both in the device, medical, you know, what we've considered the medical device context, and in the context of drug development, this meeting is a great example of the very intertwined nature of those two paradigms. Those areas are working together, I think, a lot more closely at FDA. Your biomarker in a clinical trial, in your companion diagnostic, the differences between drugs and devices issues for some of these regulatory areas, for some of these drug development areas, I think become less meaningful from kind of a core scientific perspective over time. So just to give it a concrete example of where we're seeing this type of convergence, FDA released 2 draft guidances early in January. One covers AI in drug development, or actually AI uses throughout the drug life cycle, and the other draft guidance addresses the kind of fine-grained contents of regulatory submissions for AI-enabled devices. So we have a device guidance and a drug guidance in terms of the old paradigm that really distinguishes those two worlds. I mean, I just can't overstate the distinction between drugs on the one hand and devices from a regulatory perspective, an FDA operational perspective, and personnel perspective, and so forth. But the issues that are covered in these 2 draft guidances have a lot of overlap, including just fundamental questions about, reviewing models or reviewing training data and evaluating more complex AI uses. So I'm optimistic because I think that a lot of work is going into this from a regulatory perspective, but who couldn't be anxious a bit given the pace of technology we're seeing.
Great. And Lauren, I want to give you a chance to weigh in here from a company's perspective in terms of how are companies interpreting and applying AI-related regulatory frameworks in your space, and where are the gaps and areas that need more effort and clarity?
So I think, this is a DC-based crowd, right? And so, there's a lot of conversation about AI. What should we do about AI? What's the right framework for AI? And I think, it's important for us in this room to, remind others that, we're already heavily regulated. We talked about the device framework. We talked about the use of AI in drug development. The data, the healthcare data that we use to, train these models and develop them is, covered by HIPAA. So there's already a set of rules that we're operating under. And I think FDA actually in issuing these really specific AI titled and directed guidances is doing a good job of talking in FDA terms about these things. Because we wouldn't want to get swept up in some of these larger non-healthcare efforts and concerns about AI. So given that we're operating in FDA's current authorities, right? We have, when we're clearly a medical device, Tempus has a cardio algorithm that is software as a medical device. It's been launched, gone through the process, and there's a number of similar algorithms like that we all understand what the rules of the road are. It doesn't mean that the sort of interactions don't raise novel issues or things that may not have been considered, because you're really taking large swaths of data and looking at them, potentially in a different way than the more traditional medical products. So, we certainly have found our interactions, to raise, new questions and, constant engagement when you're bringing through these kinds of products. But I think in my experience dealing with FDA, those kinds of products and interactions actually lead towards guidance development. I was in a center, so I have a center bias, as does Joe, where you say, oh, we're dealing with these kinds of products, let's figure out a way to communicate more broadly through guidance. So I'm hopeful that the agency actually will continue to do that with the assistance of product developers.
Great. So
can I just,
oh, yeah.
Just to emphasize one thing Lauren said, I think, and again, just another theme to explore, clear rules of the road. I think it's so important to complement the kind of more kind of high level or even scientifically focused guidance development and discussions around regulatory science with the type of clarity that we've had for a very long time on the FDA side. I think it's almost easy to gloss over the level of specificity that we have in these different regulatory pathways that Lauren was alluding to. We have to keep an eye on making sure that we are mindful of those regulatory categories. They're updated. That the specific regulatory items are updated over time. I think that this is there's a question about how much resources to invest on the agency side in regulatory development, and making sure that even guidance documents are grounded in some of the core regulatory concepts that we have, and that we kind of help perpetuate that level of a discussion as we're advancing some of these regulatory science issues. So anyway, that's my hobby horse, but I wanted to mention that.
Great. So we heard a lot earlier on the other panels as to the examples of just how iterative AI is in nature, and that obviously poses sort of unique questions for regulators in ongoing oversight of those technologies. So for Joe and Rohini, given AI's iterative nature, what regulatory approaches or mechanisms could enable that really critical oversight, but don't put barriers to innovation and change in the way? And are there any specific models that you can point to that might be helpful in this space?
Well, I think that we had there were some allusions to predetermined change control protocols earlier in the meeting. I think that I'd be super eager to hear what Rohini and Lauren have to say from the device perspective. I think one aspect of the just to ground the conversation in kind of concrete regulatory recent regulatory developments. The guidance on the uses of AI in drug development has a section that addresses so-called life cycle uses of AI. Maybe these are uses in the context of pharmacovigilance, where there's an ongoing use of the AI that's distinguished from a kind of static one-time use of AI in the context of a biomarker in drug development, for instance. Maybe it's a manufacturing use of AI. And I think that the level of detail in the guidance, I think, emphasizes a core point, which is that FDA is fairly early in evaluating how to mitigate or measure and mitigate risk for AI tools that may evolve a lot over time. I think that there's more clear mechanisms on the device side to address updates to AI, but when we think about the challenge of incorporating AI into processes like drug development, I think one of the key challenges is how do we deal with AI that is not a static tool. It's not a slide rule or a specific assay that we know very well what the performance is. How do regulatory teams grapple with the fact that it may evolve over time? That's an outstanding question. I think it's one of the core outstanding questions.
I'm definitely now realizing I'm in between two people that were at centers, and so my perspective really, it's funny because I approach a lot of things similarly as somebody that spent a ton of time on Capitol Hill, or the way like maybe even the difference between like what a White House would want to see in the context of all of this. But there's always that tension. I think that, so if we're thinking about the way even lawmakers are observing all of it too, there's always that tension between people trying to make sure that things are safe, that, there's going to be questions about, well, where is the human in the loop in this context? Is it the doctor? Is it the, the practitioner? Where are they involved in these frameworks in terms of, and then, one of the other things, at least in the healthcare side, when we deal with healthcare systems, a lot of it is, does this fit into our workflow or not? Because if it doesn't, we don't know how to use it. We can't, we have too many things to do. And so there's a lot of those conversations, I think, on the actual product side that people are thinking through. But there is a real conversation because of the nascency of where we're at and all of this about how to, I think lawmakers are probably most excited about the breakthroughs in the medical space comparative to other sectors. When they talk about AI, there's a lot more fear about just when you're purely talking about whether it's in the tech space, what are the things that these large language models can do. And so there's a lot more, caution there. Of course, there's caution, I think, in the healthcare side where they're thinking through, we want to make sure people are safe, that they're, but at the same time, the idea, and I think this is a bipartisan interest, but the idea that these things can lead to better cures for people, faster cures, better treatments, all of those things are really exciting in terms of where people could see us a decade from now. And so there is going to be pressure to also make sure that we are addressing the needs of people that are looking for breakthroughs in medicine. I think one of the challenges, I realize that it is an FDA-heavy conversation, but there's no question we're going to have to really reevaluate the CMS and payment side of the world. Right now, even the current guidance of, essentially widespread use of AI, in order to, essentially what it means is that you have to be able to show some version of widespread use because the legal statute in Medicare is around medically necessary. Is this something that people really need? You have to justify that versus an FDA standard of safe and effective. So you have a lot more products that are waiting for a medical code. They're waiting for, they have to just show a different justification on the payment side. Both of those things are needed to truly get to the patients. And so I think, it's certainly not an easy thing to grapple with because particularly the world of, I think, Capitol Hill, others, companies, as they literally sit through and have to think through both FDA and CMS and access to, getting access to their patients. You have to sort of go through all of these steps, and this is something that it's definitively one of the things we'll have to grapple with in the future as this evolves.
Lauren, thoughts you want to share on this question?
I think that there are, I appreciate your comments very much about, the challenges in seeking reimbursement, because these tools they're tools, they can be so new. Yes, some of them fit squarely into the medical device framework and are more clearly defined, but the potential and what's coming, our system was not set up to anticipate. So, I think thinking through the challenges of the best reimbursement pathway is, something that needs a lot of, input from, groups like, Friends of Cancer Research, but also the developers to sort of explain how we're thinking about the value that this can bring to patients. I think, we're going back and forth on, clear rules versus not clear rules, but I think we should also recognize that AI has become so accessible, right? Like just using it on your phone is incredible. Our kids are all, able to use it. And so when we think about it being incorporated in health systems and by providers, outside of the traditional, like I'm a device developer framework, I do think we need to, look at FDA policies around clinical decision support, because it has the potential to sweep in, I think, more than was anticipated when we were thinking about, that legislation and those guidances more in the traditional device framework. Because, AI can be incorporated, into EHRs and, within a health system based on, physician experience or expert experience. And that's not how we traditionally thought about products coming to FDA. And so when I think about the two big things we really need to focus on, it's payment and where the line should be on clinical decision support.
So shifting gears slightly, I think when people talk about AI tools in general, one of the most oft-cited concerns has to do with bias. And you saw FDA grappling with that a bit in the two recent guidances that Joe just mentioned. So for all of our panelists, what measures are needed to account for bias in AI tools and ensure transparency in their design, training, and implementation? We can start with Joe.
Representative data. I think preaching to the choir here, but I do think maybe that there is, number one, I think that there's a lot of noise around this topic right now. But I think we all know that it's extremely challenging to get good data sets. And so I think, this is clearly not just a regulatory or clinical or biomarker development problem. This is a problem that relates to the generation and accessibility of data broadly in the healthcare system and beyond. And so I see because of the extraordinary need to make sure that AI tools are developed using data that's appropriately representative from a scientific perspective, I think we're going to see only increasing pressure on data availability. And data availability, I think the data is the fundamental challenge for all of us who are working in this space. And I think that it's not just on the development side, obviously, but on the monitoring side for certain types of tools. And I think my take is that we just need to all acknowledge how challenging some of these data generation and evidence generation areas are.
Yeah, I think Joe's right. You can sort of see this issue cutting across so many different aspects of healthcare, aspects of, across sectors. It's not like you wouldn't have the same conversations about in like financial services or even in the technology space in terms of, what is the data that these models are being trained on. But I think it's more important to, to really make sure that our regulatory bodies are grappling with, as people are, as we get to a place where people can do real-time monitoring, what does that representative data look like? What is the data sets that they're using? It has to feel like everybody is on board with these issues in and of themselves, because I think, I think it's natural to say that there's going to be spaces where regulatory bodies feel more comfortable with, right? Like imaging or pathology as an example. But when it comes to what is it being used for, let's think about some case studies. Are we telling certain groups of society, you know, that this tool is going to be used, and now you know that you have cancer, things like that. We don't want that to be wrong, right? So we can all agree on some of these things, you know, no matter the tools. You could go through a lot of different use cases where, you know, maybe whether it's gender, race, geographic diversity, all of those different aspects of it, we have to be thinking about that across, you know, both regulation and also patient access too. Again, you know, in the second part of it, not just making sure that we're looking on the regulatory side, but making sure that patients are really at the center of this. How are they getting access to this, and making sure that, you know, people can all benefit from the product itself.
Yeah. So, you know, we think through this in terms of, you know, transparency about, you know, explain what we're doing, how we're doing it, why we think it's, you know, working, and also, and, you know, what are the data we use, and what are the inherent limitations in that data, right? Like we're limited by the data we can generate out of the healthcare system, and we need to recognize in situations where there may be limits on, you know, the patients that were enrolled or the subpopulations, you know, that are available in these data sets, and identify those limitations, and then what are we going to do to address it? You know, are we going to sort of further validate the model in different ways so that we can address what we know are going to be potentially some limitations in performance based on the data that were available for training? So I think, you know, being very open and about these issues and these conversations, and, you know, being transparent about how we are doing this, and, you know, where we think there may be gaps in the data, and then what can we do to, you know, address them, you know, as part of a learning healthcare system generally, but also, you know, we always want to improve model performance, and, you know, look at other ways of incorporating data to help, you know, the broadest spectrum of patients we can. So you know, I just think like being, you know, part of the conversation about this with regulators, with patients, with the users, and, you know, explaining what we're doing and what we're developing is, you know, the way to start going about this.
Great. So we heard today on the first panel about the important role that common reference data sets played in identifying potential differences among tests. Again, for any or all of our panelists, how important do you think that the availability of these reference data sets are, and is there a role for independent evaluation of tool performance using these reference data sets?
I'll just make a quick observation, which is I was fascinated by the first presentation and the work the working group has done to look at the opportunities for model developers to come together and evaluate and essentially enhance their models with a common reference set. That seems like such a remarkable opportunity to improve tools in a collaborative way. So it seems like there's a lot of promising things to learn from that specific project for this area.
Yeah, I mean, Lauren, I'd be interested to know what you think. I think, you know, we have to get to a place where, you know, we are thinking about, okay, how do we, going back to Joe's earlier comment that there's a clear set of rules of the road, and I think that the reference set, that kind of data all helps us move in that direction. So I think that any additional clarity for people is always something good that allows people to move faster.
Yeah, and I also think of it in terms of unlocking siloed data. Unlocking and connecting siloed data, I think, would also help further these efforts overall.
Great. Can any or all of you again talk through whether there are certain use cases or applications of AI-enabled tools that would benefit from modified policies or regulatory approaches?
I think that there, were good conversations around, modernizing the framework for diagnostics generally, and, being sort of a student of the medical device laws. It's not a framework that has been modernized and revisited potentially as much as it should, considering the pace of technology. And so I think some of the ideas that came out of the discussion on diagnostics modernization would be very helpful on these technologies and AI-based tools, because similar concepts apply. You're dealing with lots of data inputs. An example that's often given in diagnostics is sequencing and how much data is generated. And then when we think about it's constantly developing and getting better, and maybe you don't need to come back in with every change or every new submission. There were things about looking at technology in those conversations that would make sense here as potential ways to make sure that these devices are safe and effective, but also make the regulatory process more efficient.
Yeah, I mean, use cases are always, very they're very specific to kind of what's on the market, what are people experiencing right now in terms of access to the product. I think one of the biggest things we can look to is some of the tensions that we're going to see pushed up against. And so, people talk about truly, I think some of the gaps people are going to have to really put some time and effort around is this question about liability and what it means to be FDA approved and how it works in the health system context, or how it works with the patient and provider relationship. HIPAA is one of our, just sort of taking a step back, HIPAA is one of our sort of last standing privacy laws to date in the United States. I mean, you look across other sectors, there's not this one really landmark law that's supposed to say, well, we make sure that we protect your patient data, all of that. And now when you go to anywhere, the number of forms you have to fill out just to make sure that and there's consent that your data is being protected. What does that look like in the context as these AI conversations evolve? Does that mean a patient has to fill out 40 more forms? How does that really translate into the world of how the workflow conversation we were talking about? And this conversation about human in the loop, I think that, the decision about, where a doctor is or a nurse is in these processes, that will evolve, because people are already, they're uploading their data to different large language models now as we speak. And so, and I think you'll also find, some of the things that we see now, even with health insurance companies, and some of the things that you're reading about in the news are oftentimes things where insurance companies that were able to make part of the decision didn't need to have physician sign off on it, and that led to a number of bad outcomes. And so I think there will be different parts of our healthcare sector that are rushing to kind of solve this for us, whether we like it or not. And so we have to stay very vigilant about the future of these use cases, some of the grappling with the liability like I talked about, and then also, again, getting back to where is the patient in all of this, and are we adding burdens? Are we making sure that they have access and a voice in all of this is really important.
And just before Joe jumps in, just want to say that in a couple of minutes we'll start with the Q&A. And again, as with the other panels, the microphones will be circulating. So tee up your questions, and we'll let Joe respond to the question about use cases.
Probabilistic models in various stages of drug development, including to supplement or maybe ultimately replace non-clinical data. I think these are enormously important opportunities. I think regulators acknowledge that. But again, I want to get back to the original challenge that I mentioned, which I think is that we have a very we being our regulatory system has a very well-established kind of role for different types of disciplines and for different types of uses of evidence and for different subject matter experts coming together and making these decisions. There isn't a seat at the table for the AI kind of probabilistic model tool. So we either have to decide whether we're okay kind of replacing our existing sets of tools, our existing assays with AI augmented assays, or maybe looking for ways to use AI more expansively. And I think that it's going to be that latter. There's going to be a lot of pressure towards that latter approach. Meaning that I think that we have to. Are there rules of the road that need to be updated? I think that this is where I would make a pitch for lawyers being part of the process, because I think that there's a lot of, as FDA would emphasize in guidance documents, there's a lot of flexibility in the underlying regulations at FDA, but there may be places that need to be updated, and I think that's really important to look at. So a lot to work on.
Great. Okay, are there questions from the audience?
Thanks for coming back to me this time. So I'm Felicia Flowett, also from AstraZeneca. Nice to meet the other colleagues I haven't met. Relatively new to the team and on the policy side, so this panel is super interesting. My question is back to the patient piece of this, and what kind of outcomes measures do you think regulators would be looking for in terms of patients reporting that the diagnostic or the proposed diagnostic method works for them? And also, are you aware of what plans they have to consult patients? Like the FDA is very good at consulting patient advocacy groups, for example, on drug development. I'm not aware if they have any plans related to devices or AI.
Lauren, you want to
You know, there was a few years ago, CDRH had a public meeting about how, how patients felt about AI. Like, it was really interesting about like their trust in AI, what their concerns were about AI, and there was a docket. So, that's one that I remember there. But it's an interesting question, right? Because I'm familiar with patient-reported outcome tools and validated instruments, and yeah, like I think we would have to update some of them and think about using them differently in the context of these different tools. And so, I think that usually happens alongside product development, right? Because we're, product developers, drugs, devices, we're always looking for, feedback and that, works as part of the regulatory process. So I've seen those tools actually develop alongside drugs and devices, and I would expect that to happen here. But I think you're right that they're going to start looking a little bit different.
Lauren or Rohini? Joe or Rohini?
She wants you to go again. I think that, one of the things when we haven't raised yet so far is like also when we think about the patient, to your point, yes, obviously I think, I have been in a number of different conversations where people have brought together health systems and they brought together innovators and people from companies, and then at some point people look around, they're like, is there anyone representing patients? And so it is something to make sure that is like, that there's a patient voice always at the table. But what's also really important too is that we are actually having this issue across, it's not just healthcare. I mean, there's so many different things when, the way companies, when they talk about it, whether it's in the venture context or all of that, it's the user experience. They want the user to have a good experience. It's not always about the protection of the user. It's not about what are their rights as it pertains to this. Now, every sector has different laws and regulations in place. But I do think that's where we have to look to, Capitol Hill, that they'll be involved just like they were, one of the bills a lot of us all worked together on was Cures 2.0, which was a big part of it was making sure the patients had more of a voice with FDA, and FDA thought through how to better incorporate patient voice into this context. So I think that conversation between Capitol Hill and any administration is going to be important, and also states. I mean, I do think, not just healthcare, but I think you'll see a lot of state legislatures and attorney generals also be monitoring around the use of AI and in the context of consumer protection.
Any thoughts, Joe?
Well, I would just go back to the importance of the human-AI interface, and if we're really thinking about AI-oriented diagnostics, I think that the question is how to incorporate, when we think about incorporating the patient perspective, how to incorporate the patient as part of the context of use for the AI. I think we're starting to develop the right types of frameworks, like in the FDA draft guidance for drug development, about thinking about context of use. Patient is obviously part of the context of use for many tools, and thinking about how to incorporate that into a more formal framework is really important.
Great. Other questions? Where's the mic?
Ellen has one.
Thank you very much. I'm Maheen Khatami and retired from the National Cancer Institute. Thank you for the change of heart, allowing me to say a few words regarding AI. My question or concern is if you are putting wrong information, because I've done an invention in Biomarker and published it in 2006. So I know a little bit about the topic. if you are putting the wrong information, how do you expect the AI to make it right? Because the data that we have, there are a lot of false flags based on false foundation. So we are not getting to the right answer, particularly if we have not done the initial research in cancer systematically. You cannot get to the right answer even with the help of AI. That's my concern and question. Thank you.
Thank you. Yeah, and I think we talked about this a bit earlier about just the importance, for instance, of the common reference data sets and making sure that the AI is trained on consistent and appropriate information. But if anyone wants to just offer any other additional quick thoughts on that, I know we got a really interesting question from the online community, so I want to make sure we get to that too.
I think even in the broader AI conversation, there's this phrase they use, which is garbage in, garbage out. So there is this acknowledgement that, you know, people have to work really hard to not just on things called hallucinations, which is, you know, where you're getting wrong information, but also really back to that conversation we just had about data and, you know, where are we getting the data from? What does those data sets look like? Who has transparency into them? And so those are all part of that, I think, broader conversation.
So Rohini, maybe you can start on this next question that we got online, which is how do you think that the current political environment is going to affect the regulation and growth of AI in healthcare and drug development, and what can we do to overcome any of those obstacles or opportunities?
Oh, is it a group therapy conversation? Listen, I always sort of take, as somebody that's spent the vast majority of my years working in a bipartisan context, I think there's a tremendous amount, you know, when I look at Capitol Hill, a tremendous amount of people all trying to get educated around these spaces around artificial intelligence. And I think FDA's mission of, you know, trying to get safe and effective products out to people is, you know, is still very unchanged. I do think, you know, of course, I think across sectors, there's no doubt that any change in political climate changes the temperature in terms of companies trying to figure out what does this mean for them, and also just everyday people trying to, you know, understand all the changes that are happening. But what I think is really interesting about AI is just that we are at a very historic point, I think, in society in terms of things are just moving very fast. In terms of access to, again, I think people talked up here about, you know, just being able to use AI, like your kids are using AI, they're using it to upload so many things. People are literally trying to find their own cures in terms of uploading their own patient data and trying to ask the AI to help them analyze so they can either ask better questions of scientists and physicians. So there is a lot of people pushing, and there's also companies that are really trying to say, you know, if you help us raise this capital, like we know that given all the advances that are moving so fast that we can really deliver for patients. I don't think any of those change. I think that some of the things we talked about are still going to be areas that you'll see both a bipartisan interest in, and I think regulators will still be grappling with some of the same challenges that we've talked about up here.
Great. On that note, I think we need to wrap it up, but thank you to our panelists, and again, thank you to Friends of Cancer Research, and I'll welcome Jeff back up on the stage.
Great, thank you guys. Really great opportunity to hear from you. Thanks for sharing your expertise with us today. No doubt a changing environment where a lot of the issues that you've raised today will continue to take shape. So as we wrap up the meeting today, I want to thank everyone in the room this afternoon and online for joining us. It's been really helpful discussions today toward informing and advancing our work as a community. With all of the new data and new ideas, this is really just likely the beginning of a lot of work to come. So many thanks to all of our speakers today and the working groups that provided their time and expertise to build these projects and advance these ideas forward. And certainly thank you to the team at Friends of Cancer Research who made this meeting and all of the work possible. Your work continues to be outstanding and is greatly appreciated, so thank you so much. Please, moving forward, take the time to visit our website. Hopefully you'll find a lot of the material presented there today, as well as some upcoming events, many of which hopefully you've seen scrolling on the screens throughout the meeting today. Also, soon keep an eye out for a new section of our website which will be laying out some new summaries of key policy priorities, many of which were reflected in today's discussions, and hopefully that can be an added resource moving forward as well. So really thanks to all of you for joining us today. Your participation and support is critical to our organization. We thank you again for your continued partnership and participation. It's certainly greatly appreciated. Safe travels, and we look forward to working with you on additional projects throughout the year moving forward. Thank you.